Oh, we can start? We can start, okay. Okay. Distinguished guests, participants, ladies and gentlemen, good morning. It's my honor and pleasure to welcome you all to this capacity building workshop in clinical trials that is taking place here at the University of Abuja, now Yakubu Gowon University. Now, this program is a 12-week intensive clinical trial training course that is being organized by the National Center of Excellence for Sickle Cell Disease Research and Training and sponsored by IQVIA, a leading global provider of advanced analytics, tech solutions, and clinical research sciences in collaboration with BioVentures in Global Health. And we're extremely delighted this morning to have such a distinguished panel of researchers, of scientists, and practitioners who are all gathered here today. Without much ado, we shall be kickstarting this event by introducing and welcoming the guests and participants whom I see seated here this morning. Let me begin by welcoming the Director General of the National Agency for Food and Drug Administration and Control, NAFDAC, Professor Mojisola Adeyeye, who is here ably represented by the Head of Clinical Trials, Dr. Beno Nyam Yakubu. Please, a round of applause for him. And with us this morning, too, is a renowned researcher, a professor of hematology and blood transfusion. She is also the director of the National Center of Excellence for Sickle Cell Disease Research and Training of our great university. Please join me in welcoming Professor Obiageli Nodu. And we have a team from IQVIA, whom I just recently introduced as the sponsor of this auspicious program. Ladies and gentlemen, please help us welcome the African Market Development Specialist of the Research and Development Solutions of IQVIA, Dr. Kweku Marfo. And I can see he's not here alone. He's here with some of his colleagues. We have Dr. Teve Akindi, who is also seated this morning. Please, a round of applause for him. He's actually the Strategic Site Solutions Manager, West Africa. I'm being told that we also have a retinue of distinguished resource persons, some of whom are online. On this note, let me quickly introduce to you a distinguished expert in radiation medicine with about 4 decades experience in healthcare and academia, now at the University of Nigeria, Nsukka. Ladies and gentlemen, please welcome Emeritus Professor Ifeoma Joy Okoye. And as the names come, we shall be mentioning and introducing them and welcoming them. But in the meantime, let me quickly invite Professor Obiageli Nodu to come forward and brief us on CESTRA. A round of applause for her, please, as she comes to the high table. But I just... Yes, my attention has just been drawn to this is a big omission, another distinguished personality over there, resource person who is online. Please help me welcome Professor Jennifer Den, who is the Chief Executive Officer of BioVentures and Global Health. You're welcome, bro. Yes. Maybe you can take it up from there to cover the laps.
Thank you very much. Thank you so much. I'm really excited to be welcoming all of you to the University of Abuja, this morning, and for this exciting package of learning that has been drawn up in collaboration with IQVIA, under the sponsorship of IQVIA, in collaboration with BioVentures BioVentures in Global Health. So we are delighted. I know that we are few here, but we are many more. There are more than 500 persons online, who have already joined the program. And it's going to be a wonderful 12 weeks of talk, learning, discussion, and to be capped up with an exciting paper which we will be able to showcase our preparedness for clinical trials within Nigeria and the West African sub-region. So I'm really delighted to welcome all of you this morning. So today, it's my pleasure to introduce what we call CESTA. It was upgraded to the National Center of Excellence for Sickle Cell Disease Research and Training at the University of Abuja during the last Global Congress in Sickle Cell Disease by the Coordinating Minister of Health. And CESTA, next slide, please, this was established. Yes, it was established. Okay, it was established in 2015 in line with the strategic plan of action for non-communicable diseases, because at that time it was very clear that more needed to be done in the area of coordination of research and training in non-communicable diseases. And so this, we called it the Collaborating Center, was formed, by the University of Abuja. And the center is mandated to carry out basic clinical and translational research in clinical for sickle cell disease, particularly looking at filling knowledge gaps, existing knowledge gaps, finding cheaper and safer therapies, as well as innovative ways, of reducing the prevalence and burden of sickle cell disease. So if you look at all that we are doing, the papers that are coming out of this, they are need-based. We identify problems in this area and we do research, and then we bring out solutions which are actually, going a long way to move the field forward in the area of sickle cell disease. The center also provides the opportunity for short, medium, and long-term training, that we produce the manpower that we need to be able to work in this area, to do research and to offer clinical service. And this is one example of such a program. This clinical trial. We have a concurrent training in genetic counseling that, believe me, has attracted more than 1,000 participants, within the past 3 months. So this is really what we do. We identify gaps and then we try to fill those gaps through skilled development. And the main focus is to be able to offer better patient care for our patients, patient-centered management. And you see that some of our patients work with us in research in sickle cell disease. The exciting thing about the center is that it has a multidisciplinary team of people. Within the university, we have people in basic sciences, we work with computer scientists, we work with sociologists, we work with educationists, and within the hospital, we have a multidisciplinary and diverse team of specialists that we also work with. And so this really makes it very exciting to work at CESTA. Apart from the energy, the young people who we are training intensively to be able to carry out research. So we have all of that. And then another thing about CESTA is that it works in collaboration with other institutions. So in Abuja, we have the 2 main institutions, the H3 Africa Biorepository, IHAB, and then we also work with the H3 Bioinformatics Network, the node in Nigeria. So we work with them. They are at NABDA. We also work with many institutions within the country, the institutions within Africa, institutions internationally in the UK, the US, and other countries. So these are the collaborating institutions that we work with across the globe. SESTA, the University of Abuja has a teaching hospital. That is the University of Abuja Teaching Hospital. And the good thing about this teaching hospital is very close to the international airport. And this is important in terms of clinical trials, because once your specimens, you collect your specimens, you don't have any problem with transporting them if you have to central labs. So this is very strategic. And when we have visitors, they can just come within a few minutes from the international airport. They can get to the center. So we are well situated to collaborate and to coordinate research. And also in the country, this universities, that's a trunk, a road that leads out of the capital. So the University of Abuja is a major referral center. It's 520 bed. It has over 550 consultants. They are adding them every day. So we have everything that it takes to offer a comprehensive care for patients with sickle cell disease and to participate in clinical trials. So that's the repertoire of specialists. You have them anesthetists, obstetrics, plastic surgeons, orthopedic surgeons, genetic counselors, psychiatrists, and more are getting added. So in the University of Abuja Teaching Hospital, we have over 1,814. I say over because we add them every week. Patients that are in the registry, disease registry, where you hear about more. About 569 of them are adults, and you have over 1,200 that are pediatric patients. And I say over because not all the patients in pediatrics are in the registry. So the services that we offer are outpatient. We have emergency. We have a daycare center. And then we also have a patient support group. And here with us are the leaders of our patient support group. For the adult one. And then also there's a pediatric patient support group. We have in the pediatric sickle cell program, we have a newborn counseling service. Then we have a clinical care from 6 months to 12 years. And then we have a transition clinic and a nurse-led hydroxyurea program. So this is, a comprehensive care that is being offered to patients in sickle cell disease. Also, in terms of the services themselves, we can do exchange blood transfusion. We have an apheresis machine. We do a cell typing and allo antibody screening. Even though it is not routine service, we're hoping to make it routine. We have it in the research, as a research going on. We have a very good non-invasive cardiovascular lab, and the center, our hospital, I think is the epicenter for research in cardiovascular medicine in Nigeria and also beyond. We have a neurology clinic, leg ulcer management, genetic counseling. We can do TCD. We have various TCD machines now available, and we have complete facilities for diagnosis of sickle cell disease. So these are some of the facilities for care. And this is a research, laboratory. And then we are very proud of the newborn screening laboratory, which we just launched recently. So that laboratory is actually a reference lab for newborn screening in the country. It's the American Society. We are working in collaboration with the American Society of Hematology. The interesting thing about CESTA is that it is a nexus, for uh, all of the major research that is going on in sickle cell disease in Africa. So we have the Sickle Pan-African Research Consortium, which we are carrying out in conjunction with 6 countries in Africa and a data coordinating center in University of Cape Town. And then we have the Consortium for Newborn Screening in Africa, which is by the American Society of Hematology, which is a demonstration project to showcase or tell African governments that it is possible to do newborn screening. And then we have the very exciting patient. So that consortium is in 7 countries, as you can see on the map. And then we have the patient-centered management of sickle cell disease, which is a big implementation science project in collaboration with the Liverpool School of Tropical Medicine, Kwame Nkrumah University of Technology in Kumasi, and then the University Teaching Hospital in Zambia. And here is really exciting what is going on, because we are looking at the care that is being offered to patients according to standard guidelines. We have chosen 3 with regard to blood transfusion, hydroxyurea utilization, and penicillin prophylaxis. And the other part is a group in the community that is looking at why patients are not coming to care in the hospitals and finding reasons for this. And then the 2 groups are working, different iterations of identifying problems that they want to solve, looking at the solutions, working the solutions, and evaluating. And this is going on. And then the other one is the Sickle Cell Genomics Network of Africa. We've actually concluded data collection and doing the sequencing at the moment. It was led from the University of Ghana with Tanzania, Nigeria, in the several centers in Nigeria that participated in that. So we have a number of ongoing research in CESTA. I will just, list them here. One thing I like about the center is that we are doing large-scale implementation science projects. When we did the first level evaluation of the patients in our registry back in 2019, we found out that the level of hydroxyurea utilization across Nigeria was just 13%. We were shocked, and we tried to find out why this was so. A number of papers came out of this, but our response to that was an implementation science project that is aimed at addressing the barriers that we found, which is that most of the barriers were physician induced. Physicians do not want to use hydroxyurea. So we are looking at that at the implementation science project we call Accelerate, seeking to increase the adoption of hydroxyurea utilization by task shifting to primary healthcare centers. So we are doing this study in 16 centers across the country. So that's a very exciting study that we are doing. And then, of course, we have the implementation science on newborn screening, where we are trying to find out a cost-effective approach to newborn screening using dry blood spots. So this we are doing not only in Nigeria, but across the consortium. We also have a study on observational study in malaria, just to find out the patients with sickle cell disease, look at them and see. And then that is a foundation for the chemoprophylaxis study that we want to do in malaria. So these are some of the things, genomics. We are also doing genomics study with King's College and with Inherent. Where am I pointing this? Okay, so that's Sickle Pan-African Research Consortium, which is a major infrastructure grant by the NIH. And the consortium is what we set out to do about 8 years ago was to build the infrastructure for future studies in sickle cell disease by building a disease registry, which is now almost 40,000 patients across the 6 countries, over 10,000 patients in Nigeria. And then we also developed uniform standard of care guidelines. We built skills in research and healthcare, and then also skills in database management. And it is some of these things that have now prepared us to participate in clinical trials. So the registry, so that's the Sparkle registry. If you look at that map of Nigeria that is in green, that's a source of joy and pride, to us in Nigeria, because you can see the many centers in the country that are participating in that disease registry. And so we have my colleagues, most of them are online, today. These are all the principal investigators that have contributed, to this disease registry that are now poised, to undertake clinical trials. And if you look at, the top, this one, you see the patients, the age, genotype categorization. Most of it is the SS, and then you see the few that are sickle cell beta thalassemia, and then you have the SC. But if you look at it, you will see the age categorization of the patients, as well as where the hospitals where these patients have come from. So having the SparkNet registry really helps with feasibility for clinical trials, because no matter what the sponsor wants, you can easily go in and check. The patients are being prospectively followed up. So you can check, from the registry, pull out those who are eligible, and it helps with screening. And then the PIs are highly skilled and their team members are getting trained. So we have organized this clinical trial in order to help the sites to become involved, in clinical. About 6 of those sites are currently, but we need to have more, because we believe that it's not just enough to be the country with the highest burden of sickle cell disease. We should be part of providing the solution, right, to it. So at the end of the course, this 12-week course, we know that the participants will understand the fundamental concepts of clinical research and trials. They will be able to differentiate between various phases of clinical trials. They will appreciate the ethical, regulatory, and scientific framework for trials. And then they will be able to describe who the stakeholders in that space are, and then they will understand how to. But this course is very practical, and we'll be speaking, in a language that people just giving tips. So some of the faculty are those who are currently, involved in clinical trials, and we'll be speaking out of our experience and just sharing so that we are all ready and we'll be able to do that. So we have an interesting, let me go quickly to the last and then back. So this is the program, and we have put the program for you. It's called the Jotter. And so you look at the program. So we have an exciting array of work that we'll be going through. And then if I go back, this is how we got to this place. We were trained, by the NIH Center for Clinical Trials back in 2011. by Professor. She led a crew from the NIH to come to this institution and train people in 13 centers across the country. And then it was followed up by Professor Clement Adebomowo, who got an NCI grant just to train us in clinical trial. And then when we wanted to launch forth more, a few years ago, Dr. Jennifer Dent, the CEO of BioVentures for Global Health, trained us intensively over a period just so that we can be able to do more of this. And then we had Rose Onia, who was the one that started the feasibility site assessment, to get us involved in the recent clinical trials, were like 5 of them. And then in June, we had IQVIA coming, and you remember that panel where we were able to really discuss, clinical trial leading to this. So we are very grateful to IQVIA, and then of course grateful to my amazing team, Ancestor, for making the work that we do so exciting. Thank you.
Wow, thank you so much, Professor Nodu, for that comprehensive and insightful brief on Sestra and SparkNet. A brief has not only given us a clear picture of the vision of that institution, but it has laid a basic foundation for us for our discussion ahead. Please, let's put our hands together once again to appreciate her. Well, I'm being told that the acting vice chancellor of this great university will be coming maybe later in the day, if you can find him out from his very busy schedule. And it will be our pleasure to welcome him when he comes to this forum. It's interesting that we're making progress. Next, we'll have to invite Dr. Kweku Marfo to give us a brief on IQVIA. Please, a round of applause for him as it comes to 7.
Hello, everyone. Thank you so much, Prof. And I think as you laid out, it has been a journey, and certainly IQVIA appreciate the opportunity to be part of this journey. I'm here today not alone, but represented with my colleagues, Dr. Teva and also Dr. Ola, and IQVIA is pleased to sponsor this event. I also have a few of my other colleagues who are also joining us online. And also during the course of the 12 weeks, there will be several of us supporting Sestra in terms of delivering this intensive 12-week capacity building program. As Prof indicated, Nigeria has the highest prevalence of sickle cell disease. So it's really important as we're thinking about innovative solutions to address this disease that Nigeria plays a critical role in this. In the next 5 to 10 minutes, I will just give you a brief intro about IQVIA and in terms of some of the opportunities that we have for potential collaboration with University of Abuja Teaching Hospital, as well as other potential partners. So as an organization, our aim is really to power smarter healthcare for everyone. This means that accelerating innovation for a healthier world. And as we're talking about sickle cell disease, on the cusp of bringing new innovation, we provide professional services to life science organizations and also healthcare organizations in 4 main domains. The first one being public health, digital health, and then also access to care. And then finally, on the clinical research side, which I'll be focusing the most of the discussions around. So from a clinical research standpoint, we've been we've been partnering with organizations over the last 30 years in Africa. Some of the areas that we've been able to deliver is from an extensive clinical and regulatory expertise across 25 countries on the continent. This has yielded to extensive relationships in over 1,200 sites. And also, we've been able to enroll over 300,000 patients into our clinical trials. Our experience have expanded from infectious disease to hematology to oncology, as well as cardiovascular disease, which are some of the diseases that we've seen as prevalent on the continent. And I think as Prof was indicating, I think University of Abuja also have expertise in all these diverse disease areas. Beyond the clinical research, we've also supported some of the first commercial launch of clinical trials in non-communicable diseases in areas like oncology, as well as also some medtech solutions on the African continent. We have over 1,500 Africa team members across diverse roles, and they all vary in terms of the level of experience on the continent. And then beyond the clinical research, we are also innovating through partnerships with offerings such as clinical academic research enablement model, which allow us to deliver to support investigator-initiated trials, academic-related research. In terms of our staff, as is indicated here, we have team members across the continent. And if you look in blue, some of the areas where we have in terms of our head offices. Here in Nigeria, we have a team that are based in both Lagos as well as in Abuja. And as you see, in terms of the multitude of services that we have, we offer different services relating to clinical research, both from east, west, or southern part of the sub-Saharan Africa. Indicated here is the diverse team that is supporting in terms of some of the work that is being done. The team in sub-Saharan Africa is led by Chris Obwanga, and across the different spectrum of the clinical research lifecycle, we have key team members supporting from regulatory to quality, as well as also site management. And during the course of the 12-week intensive course, you'll be hearing from some of these team members who will be sharing their expertise in some of these respective areas. Beyond the support that we provide in clinical research, we also contribute through thought leadership in publications, as well as also research papers. And I've listed a few of some of the publications and research papers that we've been part of, but actually most recently, it's a research paper that a team released on building Africa clinical trials, leveraging on sickle cell disease experience to actually expand into non-communicable diseases. And then finally, just a few additional in terms of thought leadership or publications. And if you're interested in any of these publications, we will be happy to provide some of these to you beyond or in addition to the services that we provide in clinical research. And then finally, I think as the saying goes in Africa, I think if you want to go far, we go together. But recognizing in terms of the significant burden of both non-communicable diseases and communicable diseases on the continent, we are not only looking to go far together, but we're also looking to go fast together. So as an organization, given the fast growing, changing healthcare landscape on the continent, we've set an aim to actually increase Africa's clinical footprint on the continent. Currently, unfortunately, we see only 4% or less than 4% of global clinical trials are conducted on the continent. We are aiming to increase this number to 10% in the next 10 years, and we believe we could only do this through partnership like this, through initiatives where CESTRA as well as BVGH is putting together to build capacity for clinical research on the continent. And looking to do this effort, certainly shaping the ecosystem where we're working with partners like CESTRA to help build capacity, investing in countries to make sure that they have the requisite expertise to be able to deliver on some of these clinical trials, focusing on therapeutic areas that are of relevance. I think we're talking about sickle cell disease now, but I think there is also opportunities in other non-communicable diseases or other disease areas such as oncology, such as cardiovascular disease. And then finally, ensuring that we are adapting new innovation to allow us to deliver flawlessly and also to be able to deliver for patients that we care most for. So as an organization, we're certainly looking forward to these type of engagements and these type of partnership in order for us to be able to not only deliver, but also to differentiate and also to ensure that innovation gets to patient as quickly as possible. So with that, I just want to say a big thank you. We will be here throughout the course of the day. And as I indicated previously, we are going to be part of also the next 12 weeks. But should you have any questions about the services that Akivia has to offer or anything in general, please don't hesitate to reach out to one of us or some of our team members that are virtually online with the team. Thank you so much.
Oh, thank you so much. Thank you once again. We just had another brief this time around from one of the sponsors, Akivia, right? Sorry? Okay, the sponsor, not one of them, and that's a key. Yes, Akivia, and the presentation was made by none less a person than a specialist there, Dr. Kweku Marvel. Please, let's have another round of applause for him. And next, we shall be going to our resource, persons who are online. And this time around, I've been told that we have a 10-minute video presentation, which is kind of a brief on bioventures for global health. And the Dr. Jennifer Dent, the Chief Executive Officer of this organization, I guess she's online. But before we probably call on her for further insights into the video we are going to present, please, let's call our, I think you're set for the video presentation over there, right? Please, let's go ahead. So we shall be watching very interesting and insightful brief on this organization for the next maybe 9-10 minutes. Let's have it.
Called AAI, or the African Access Initiative. AAI is focused on improving cancer patient outcomes in Africa by establishing affordable access to high-quality cancer medicines and technologies, building clinical oncology capacity, addressing the cancer data gap in Africa through clinical trials, and expanding awareness of cancer in Africa. We partner with governments, hospitals, pharmaceutical companies, other private industry partners, nonprofit, academics, and more to implement our programs. In particular, we've partnered with the Ministries of Health and cancer hospitals across 8 African countries, namely Cameroon, Cote d'Ivoire, Kenya, Lesotho, Nigeria, Rwanda, Senegal, and Tanzania. Within each of these countries, we have signed memoranda of understanding with the Ministries of Health, and the ministries have dictated which hospitals we should partner with and to strengthen and support their healthcare and oncology capacity. Within each of these hospitals, we work closely with the leadership across the whole hospital, the oncology, pharmacy, pathology, nursing, and other departments to implement programs aligned with their goals, priorities, and needs. To understand those goals, priorities, and needs, we first start out by conducting a needs assessment that delves into what the hospital sees on a daily basis in terms of the number of cancer patients diagnosed and treated, as well as what the hospital's diagnostic and treatment capabilities and infrastructure are, their cancer research and clinical trials goals and capabilities, their human versus capacity across skills, as well as the cancer drugs that they use and need on a routine basis. Based off of this information, we develop programs that are customized to address gaps and fill needs with our hospital partners. In the area of cancer drug access, we work closely with pharmaceutical companies, governments, hospitals, as well as supply chain stakeholders to implement cancer drug access. We work with hospitals to understand what cancer drugs they need access to, the patients that they would treat with those drugs, and based off of that, we both forecast the drug quantities that would be needed to treat those patients, match those drugs with companies, verify those companies' affordable access prices, and then calculate the burden or the budget that would be needed to procure those drugs. Once the hospital has agreed to procure those drugs, we help to coordinate access agreements with the company or the supply chain stakeholder, and then support and manage all logistics from port to patient to ensure that products arrive in a timely manner and are delivered to patients appropriately. To date, more than 4,000 cancer patients have accessed cancer drugs through our program. All of our cancer drug access programs build upon the existing structures within a country and within a hospital. To ensure that the pharmacy, the oncologists, the nurses, and more have the appropriate capability to diagnose and treat patients appropriately, we leverage a network of global oncology experts to conduct capacity building programs. Our programs cover skills across the cancer continuum of care, everything from early detection and screening to diagnostic and imaging capabilities, nursing, patient care, treatment, survivorship, and more. We conduct these trainings through digital training programs, digital mentorships, in-person workshops within country, as well as fellowships and more. All of our digital training programs are uploaded onto our on-demand online learning platform called BVGH Learn. This allows individuals who weren't able to attend the courses live, want to rewatch the courses, or any other reason to rewatch or watch for the first time the courses, take the pre and post course exams, and receive a certificate of participation. In the area of clinical trials, we also work to raise awareness of Africa's clinical trial capabilities by using an online platform to profile hospitals' research experience, capabilities, infrastructure, diagnostic and treatment capabilities, data management, pharmacy systems, and more. And the goal of this platform is to not only highlight the capabilities across the continent, but to encourage international organizations, including PIs, pharmaceutical companies, and more, to partner with African hospitals to conduct African-led clinical trials that are for African patients. Leveraging the information we have gathered from those profiles, we also conduct clinical trial capacity building. This includes digital training programs, on-site training programs that are workshop-based, as well as individual fellowships, both virtual as well as in person. We also coordinate a variety of research as well as clinical trial consortia. One such example is the Hypo-Africa Consortium, which consists of global oncology experts across the globe that are focused on analyzing hypofractionated radiotherapy for the treatment of cancer. 4 sites in Africa have key PIs, namely the MLCC Center in Nigeria, the Uganda Cancer Institute in Uganda, Ocean Road Cancer Institute in Tanzania, and the Nkosi Albert Lutuli Central Hospital in South Africa. And lastly, we focus on improving cancer patient awareness to help downstage the type of cancers that are seen in the clinic to the more treatable stages. One such example is the development of prostate cancer awareness materials, which we worked on with the Medicaid Cancer Foundation in Nigeria. These materials were developed and then distributed in local communities outside of Abuja. Men were also engaged in presentations about prostate cancer and encouraged to talk to their physician about prostate cancer and to get regular prostate screenings. And finally, we also worked to raise the international community's awareness of cancer in Africa, with the goal of spurring more investment and support to build and strengthen Africa's response to its cancer burden. So I thank you very much for today allowing me to present, and I hope you have a wonderful workshop, and I look forward to seeing everyone on the subsequent digital training programs. Thank you.
Well, again, thank you so much for that presentation, and I thank you, this great audience, for watching this video on bio, which is a brief on bio ventures for global, health. I do hope that you found the video enjoyable and insightful. Please, let's have another round of applause. Well, we're moving forward. This time around, we're going to be taking another brief on introduction to clinical trials and trial phases. This time around, this is going to be presented by no less a person than a professor of radiation medicine at the College of Medicine, University of Nigeria, Nsukka, and the University of Nigeria Teaching Hospital, Enugu. She is also the director, University of Nigeria, Nsukka Center of Excellence for Clinical Trials, and an adjunct professor, research and innovation of the National Institute for Cancer Research and Treatment. Again, I see that she's also the Provost General Living by Design Institute. I'm talking about a renowned professor who will be presenting. Please, let's welcome on board Emeritus Professor Ifeoma Okoye, right? I hope she's online for the presentation. And before she comes, in the last couple of minutes, we have been having different, briefs and presentations from the sponsor of this program, and also from the organizer. And we just had another one from one of our resource, persons. And I do know that we have been listening very attentively. And in the next couple of minutes, we shall be piecing all of this together and saying how much takeaway we can make from that. Aside from this presentation, we have lined up for you this morning an overview of, we shall be having another one on history and evolution of the trials, and we shall be having a press conference. And I can see that some of our media persons are just joining us, this morning. I can see Arise Television, I can see The Punch, and a whole number of them who have joined us this morning. Please, let's have a round of applause for them as they join us today. We are still waiting for our emeritus professor. I don't know whether she's, online.
Yes, I'm online.
Oh my God. Well, that was a very powerful voice.
Good morning, everybody.
Good morning, thank you.
Good morning, Ma. Hello, ma. We can hear you, ma. Please proceed with your presentation.
Okay. I choose from being okay. Nice. It's me. So then it's me. Should I start?
Yes, ma, you can start.
Okay. Good morning, everyone. Thank you very much for having me. We are here to listen to an introduction to clinical trials and clinical trial phases. And I'm Professor Emeritus Ifoma Okoye. And today, I'm going to introduce you to the basics of clinical trials, what they are, why they matter, and the different types and phases that we have. This session is actually very important because understanding clinical trials is the foundation for us to appreciate how new drugs, vaccines, and treatments reach patients safely and effectively. Without clinical trials, medicine would actually simply be a guesswork. I want to believe that somebody is managing my slides because I can't see any movement at all.
Move.
So let's look at some definitions and key terminologies because I believe that those IQVIA and BVGH realize that some people in the audience may be new to clinical trials. So we'll be looking at what is a clinical trial. And I would like to start with the World Health Organization, WHO, definition that defines a clinical trial as any research study that prospectively assigns human participants to interventions to evaluate the effects on health outcomes. Now, let me explain this step by step. Prospectively means the study is planned in advance before participants are enrolled. And what do we mean by assigning human participants? It means that people are deliberately placed into groups. For example, one group might get the new medicine and another group might get the standard treatment or even a placebo. Now, interventions are things done to test, like a new drug, a vaccine, a medical device, or even lifestyle changes such as diet or exercise. When we say effects on health outcomes, we actually mean that we want to see how this intervention changes health. Does it cure the disease? Does it reduce the symptoms or ameliorate it? Or does it improve the quality of life? So in simple terms, a clinical trial is a planned scientific study where people are given treatments and researchers watch carefully to see if the treatment that is being given works and if it is safe. Now let's look at the acronym ICP, ICH, and GCP. What do they mean? This acronym ICH, it stands for International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. The ICH is actually a global body that brings together regulators and the pharmaceutical industry from regions like Europe, the USA, and Japan. And their role is to harmonize standards so that when a trial is done properly, the data can be trusted and accepted across many countries, because you see that clinical trials are done across many countries, across many populations, so that the outcome product will have the effect that it has been able to be standardized to have. So now let us look at GCP. GCP stands for Good Clinical Practice. It is the international ethical and scientific quality standard for designing, conducting, recording, and reporting clinical trials. The purpose of GCP is twofold: to protect the rights, protect the safety, and the well-being of trial participants. Trial participants are those who are recruited into the study to be the human subjects that will have the intervention method on. The GCP also ensures that the results of the trials are reliable and credible. So it is very, very key. When we say ICH GCP, we mean the globally agreed standards developed by ICH that clinical trials must follow. Now for WHO also has its own GCP guidelines, known as WHO GCP, which are aligned with ICH but focus strongly on ethics and participant protection in public health settings. So whether you hear WHO GCP or ICH GCP, the bottom line is the same, and that is that clinical trials must protect people and must produce results that the world can trust. Now, there are some other key terms that you must know when you're getting into the clinical trial ecosystem. The first one is protocol. This is the written plan of the trial. Think of it as the blueprint or constitution of the study that must be followed to the letter. It explains the objectives of the study, the design, the methods, and who can join, and what will be done, and how the data will be analyzed. Now, without a protocol, there is no trial. It is actually the very beginning, the alpha. Now we look at the informed consent. This is not just signing a piece of paper. You just tell the patient, as we used to do when we were medical students, and later on when we graduated many years ago, just before the surgery, the night before, you go to the patient and just show the patient the paper. The patient wouldn't even need to read it and just say, sign your signature here, because nobody taught us the rights of the patient for this informed consent. So it is a process. The participant is fully informed of the study, its purpose, the risk, and the risks can be from mundane to profound. For instance, if you're going to give an injection, you will explain exactly how the patient will feel, what you're going to do, how the pinprick, will hurt a bit and all that. So you must explain everything and the process. So the participant is fully informed of the study, its purpose, the risks, the possible benefits, their rights, and their freedom to withdraw at any time. And guess what? You are actually supposed to be able to give that informed consent to the patient or to the participant, if he's not a patient participant, to go home and consult with their family or people that are in authority over them, so that they will decide that truly they have been informed and they are consenting. So only after understanding all this and asking questions would the participant or the participant patient agree to join. Now let's look at the word placebo. In the word placebo, a placebo is an inactive substance. For example, a sugar pill or a saline injection. It is designed to look like the real treatment. So when you're looking at the real treatment and looking at the placebo, they look exactly the same. And helps researchers to compare results fairly so that they know whether the real drug works better than doing nothing. So it actually helps to remove bias. So randomization is when participants are assigned to groups purely by chance, like tossing a coin. It prevents manipulation, bias, and ensures that there is fairness in the study. So that's what randomization does. You are not able to favor any of the participants. You just randomize. Now let's look at the word blinding. Blinding, this is when participants, investigators, or both, that is, both the participants and the investigators do not know which treatment is being given. So it's either both, or the investigator is blinded and the other is not blinded, or the two are blinded. So this prevents expectations or bias from influencing the outcome results. So for example, if someone knows they are on the real drug, they may feel better even if the drug doesn't work. That is the placebo effect. Blinding keeps the trial honest. Now let's look at the word endpoints. For endpoints, these are the main outcomes being measured. For example, did the patients live longer? The patients that had this intervention, did they live longer? Did their tumors shrink? Did their blood pressure drop? The endpoints are the yardsticks that show whether the treatment actually worked. Now, for the word adverse events, this is really a very important aspect of the trial. Please, somebody is saying that the slides are not moving. Please, can you move the slides to align with, I'm now in adverse events, please. So these are any unwanted or harmful effects that occur during a trial. It is very important, and it does not matter if the drug caused that effect or not. Every adverse event must be recorded, must be reported. For example, a headache, a rash, or even a broken leg during the trial must be reported as an adverse event. So whether it's the drug that caused it or not the drug that caused it, it is an event that occurred during that trial. So we are now done with the definitions, and we are going to now look at importance of clinical trials. The question we want to ask ourselves and to understand where clinical trials is supposed to be taking us to is, why do we need clinical trials? Couldn't doctors just try new treatments directly? Like everybody was accusing conspiracy theory during COVID period. Now, clinical trials are the gold standard for proving that new drugs, vaccines, or devices are safe and effective. Every treatment you know today, chemotherapy, radiotherapy, even paracetamol that people take ad nauseum, once was tested in a clinical trial. Trials protect patients. Without trials, unsafe or ineffective treatments could be used, causing harm. I'm sure that in the course of this workshop, you are going to go into the details of why there was even the need to have that ICH and have GCP put in place in human research. Now, clinical trials drive innovation. Clinical trials turn discoveries from the laboratory into real solutions for patients. So clinical trials take solutions that were discovered in bench to the bedside for utilization for medical care. Now let's, an example, the HPV vaccine, which we know that is being used to eradicate the whole issue of cervical cancer. The HPV vaccine was proved effective through trials before being rolled out worldwide to prevent cervical cancer in those 1980s and into 1990s, when people were, suspecting viruses and all that as part of the process of this cervical cancer. People thought people were just theorizing. And now the world is talking about eradication of cervical cancer. So we have 4 types of clinical research. If you can go to the next slide. There are 2 main types of clinical research. Interventional studies, number 1, and observational studies. So let's look at interventional studies. In interventional studies, researchers assign participants to receive specific interventions like a drug, a surgery, or a lifestyle change. The goal is to test the cause and the effect. For example, giving one group a new breast cancer drug and another group the standard therapy, then you compare the results. That is interventional studies. Now, for observational studies, researchers observe participants without assigning interventions. They may track lifestyle, genetics, or environment. There's a big study we are involved with now that is called the Sambai Project, and it's all about the social determinants of health. And it's quite a very interesting study that is going to definitely change paradigms. Now, an example of observational study is where you are following smokers. In this particular study now, we are following the dietary patterns, studying every detail of the diet of all the participants that are in the study. So following smokers and non-smokers over time to study lung cancer risk. And of course, in environments where you have some environmental risk factors like, in Enugu, for instance, where we have coal, and coal has been associated in literature with higher incidence of breast cancer. So we are looking forward to this particular study of social determinants of health to see what we are going to unearth from that study. So to put it simply, in interventional, we do something to the participants. In observational, we just watch what happens naturally. So let's look at the 5 phases of clinical trials. Clinical trials progress in steps called phases. Each phase has a different purpose. Now, we start with phase 0, and we have phase 0 to phase 4. Now, for phase 0, exploratory, that's what we are doing there. It's an exploratory phase. Very small, first in human studies, using microdoses. Currently in Nigeria, we have not begun to receive approvals for first in human studies. Of course, I saw that NREC and NAFDAC are going to be potential players in this workshop, so they're going to give you all the details. So understand how the drug behaves in the body. That's what you want that phase 0 to give you, the answer you want from that phase 0. Now, we go to phase 1. Phase 1 is where we are looking at safety, the safety of that IP, the interventional product, the new drug entity, some people call it. So in phase 1, you have a small group, 20 to 100. The focus is to answer the question, is it safe? And another question, what dose is right? Example, checking for side effects like nausea or liver changes. And in phase 2, we are checking for effectiveness. These are, they are candidates to intervene in that disease entity. So you want to check, is it having the effect that you have pretended that it's going to have? So in this phase 2, you're dealing with larger groups, 100 to 300. And the focus is, does it work? And is it still safe? For example, does this drug shrink tumors better than placebo would? In general medicine, phase 1 would usually be healthy volunteers, and phase 2 would be patients. In cancer and HIV, and some people usually add tuberculosis, for instance, both phases 1 and 2 usually involve patients because the drugs are too toxic for healthy volunteers. Now, for phase 3 is targeted for confirmation. It's targeted for confirmation, and it involves a large group, about 1,000 plus patients, often across many countries. So they are multi-center, multi-country, multi-continents. And it compares new treatments to the standard of care. It provides definitive evidence for regulators. And then from that phase 3, that evidence you have provided as evidence for regulators will lead to an approval for utilization in human population. And then when you get to phase 4, you are now talking about post-marketing surveillance. After approval, the drug is monitored in real-world use to track long-term safety and to check for defects. Some of these long-term safety and rare side effects can occur several years after the drug is already in use, like the NSAIDs, for instance. At a time, they said, take Celebrex and the rest of them out of the market. But the patients have continued to use them. It is now the choice of the patient to insist they want to continue to use it. Apologies, they just took light where I am, so I'm a bit dimmed. So I go to the next slide, and we are going to, I believe that is where I have yes. So this is actually supposed to help you just in a one eyeballing, be able to see all the differences of exactly what I have been talking about for each of them. Exploratory, very small number of patients, explore how the drug behaves as the main goal, and typical sample size. So you have that for phase 0, phase 1 for safety, phase 2 for effectiveness, phase 3 for confirmation, and then phase 4 for post-marketing. Post-marketing patients in the general population using the drug is what you use to track long-term safety, effectiveness, and rare side effects. So there is an aspect that I would like us to use and end with. If you go to the next slide, it's going to be showing you something on pharmacovigilance. Very good. So pharmacovigilance, even we must keep watching medicines. Pharmacovigilance means detecting, assessing, understanding, and preventing adverse effects or other drug-related problems. Some of you who are old enough when Professor Dora Akunyele was the DG of NAFDAC, pharmacovigilance just literally like jumped into Nigeria and took fire. And she really, had a lot of jingles on pharmacovigilance, and it became a very popular terminology. So why? Because rare or long-term side effects may only appear when millions of people use the medicine. Reports are collected by agencies like WHO's VGBase, US FDA's MedWatch, or NAFDAC's Pharmacovigilance Center in Nigeria. So if risks are confirmed, regulators may update warnings, restrict use, or withdraw the drug. Now, always remember, phase 4, post-marketing surveillance. Post-marketing surveillance can be said to be equivalent to pharmacovigilance. And pharmacovigilance means our commitment to the patient's safety, that our commitment to the patient's safety never ends, is continuous. So in closing, if you can shift the slide, in closing, I would like to literally do a summary. The next slide, please. To summarize, clinical trials are the bridge between medical research and patient care. By understanding the definitions, importance, types, and phases, you can appreciate how rigorous and ethical the process is. It has to be robust. The oversight has to be robust. Nobody is going to bring down the standards for any group of people. Clinical trials is a robust system, and it has to be ethical, and the process has to have all this in place before a new treatment reaches patients. So without clinical trials, medicine will be blind guesswork. But with them, we protect people and advance science for the good of humanity. So I would like to end with these words that we are so happy to have an increase in interest in clinical trials in Nigeria, and we look forward to different sites being able to be part of it. Thank you very much for inviting me.
Wow, thank you so much. That was a beautiful presentation on introduction to clinical trials, which took a comprehensive look at the definitions of some terms. One of the terms, that struck was pharmacovigilance, or how was it called? And some other, terms there that she beautifully made reference to. And of course, this is quite expected from an emeritus professor of IE. Please, ladies and gentlemen, let's have another round of applause for Professor Ifoma Okoye for that beautiful presentation. And then I think we go to the next presentation, which is on clinical trial stakeholders. This one is going to be made by no less a person than a clinical researcher with over 10 years experience in managing and implementing clinical studies. She is currently an African market development strategist or specialist at IQVIA. Please, let's have a round of applause for Dr. Frida Mwendia as she comes on board for our presentation.
Strengthen clinical trials in the region. Collaborations like this are not just partnerships, they are catalysts for transformation. And together, we are building the foundation for a more robust, equitable, and sustainable ecosystem in Africa. In today's session, we'll walk you through the key players who make clinical trials possible. Let's dive in. This shows the ecosystem around a clinical trial. It's never just one player. It's a network of sponsors, regulators, sites, their personnel, and participants all working together. Each has distinct responsibilities, but the real success comes from how well they connect and collaborate. If any piece of this chain falters, whether oversight, operations, or engagement, the entire study is at risk. Strong coordination across these groups is really what drives credible, ethical, and impactful clinical research. For initiating, managing, and financing a trial. They're responsible for the over-trial conduct, protocol development, data integrity, and regulatory compliance. According to the latest ICH GCP E6R3 guidelines, the sponsors must develop the protocol, safeguard regulatory compliance, and implement quality assurance and control systems. Sponsors activate trial-specific systems only after necessary approvals have come in, and they must document all processes for data handling. The guidelines emphasize proactive, risk-based oversight and clear accountability for all delegated activities. Next, we have the CRO, the Contract Research Organization, usually a person or organization which could be commercial, academic, or other, contracted by the sponsor to perform one or more of a sponsor's trial-related duties and functions. The CRO is typically a company that manages and coordinates clinical trials. A sponsor may transfer any or all of the trial-related duties and functions to a CRO, but the ultimate responsibility for the quality and the integrity of the trial data must reside with the sponsor. Under FDA 21 CFR 3.12.52, as well as the ICH-GCP guidelines, transfers must be documented in writing, specifying each obligation in detail, and CROs must comply with the same regulations and are subject to the same regulatory actions for non-compliance. Then we have the study participant. The term subject was replaced by participant, and this was done to emphasize their active role. Participants are, after all, the heart of every trial. The participant is the only member of the team without any regulatory obligations. However, participants must ensure that they are willing to comply with the scheduled visits and investigational product regimen, as well as report any effects to their physician. They may have no regulatory obligations, but they must be willing to receive clear, timely, and understandable information. The ICH E6R3 guidelines emphasize participant centricity, ensuring that the perspectives and experiences of the participants are considered throughout the entire of the trial. Then we have the investigator, principal investigator, or PI as we call him. This is the leader at the trial site, and they're responsible for the ethical and compliant conduct of the study. Their responsibilities include ensuring participants are fully informed and that this consent is properly. recreational products. In trials with complex dosing or multiple arms, pharmacists are key to preventing errors and ensuring consistency. In the ICH GCP E6R3, it calls for robust systems and qualified personnel, and pharmacists fulfill both this by safeguarding the product integrity and by supporting trial reliability. Next, we go to the regulatory authority. Regulatory agencies are defined as bodies having the power to regulate. This includes the authorities that review submitted clinical data and those that conduct inspections. Employees of these regulatory agencies, for instance, the FDA, will have communication with and inspect sponsors as well as CROs. They could also inspect and sponsor investigational ethics committees and investigational review boards. The regulatory agencies worldwide also can and do inspect investigator sites. However, the main communication is usually with the sponsor or the CRO. The role of the regulatory authority is to protect the health and safety of consumers and patients by making sure drugs, biologics, and medical devices are safe and effective. They also ensure that product packaging and labeling is truthful and informative. Then we have the data safety monitoring boards, and these are independent committees composed of clinical trial experts tasked with overseeing clinical trials. They are the essential guardians of participant safety and trial integrity. Typically, they're made up of clinicians, biostatisticians, ethicists, and other specialists. They must be independent from the sponsor to avoid conflicts of interest. Their core responsibilities include conducting regular reviews of accumulating trial data, evaluating the risk-benefit ratio for participants, and eventually recommending whether to continue, modify, or terminate the trial. They're important because they are often involved in early detection of adverse events or safety concerns, as well as maintaining ethical standards and data reliability. They usually send these report findings to sponsors, regulatory authorities, and sometimes to the investigators as well. This ensures transparency and accountability in the trial conduct. DSMB interventions have prevented harm and improved outcomes in numerous trials. For instance, a DSMB can halt a trial due to an unexpected toxicity. Therefore, their work upholds the ethical and scientific standards of a clinical trial. Then we have the ethics committees. The ICH guidelines defines the Institutional Review Board, IRB, or Independent Ethics Committee, IEC, as an independent body. It could be a review board or a committee, and it could also be institutional, regional, or a supranational, and usually consists of medical professionals and non-medical members whose main responsibility is to ensure the protection of the rights, safety, and well-being of participants involved in a trial. The Ethics Committee is also a guardian of participant rights and safety. They provide public assurance of the protection of rights by, and among other things, reviewing, approving or providing favorable opinion of the trial protocol, the suitability of the investigator, facilities, and the methods and materials to be used in obtaining and documenting informed consents of the trial participants. The IRB or IEC provides an independent review of the research. As per the regulations, the investigator is responsible for ensuring that the IRB or IEC that is used meets the ICH guidelines or other local regulations. It is also the investigator's responsibility to know if a local IRB should be used or if a central one can be permitted. The primary communicator with the ethics committee is usually the investigator. Sponsors can communicate with the IRB or IECs where there are questions, concerns from the committee about protocol or subject safety, for instance. Finally, we have the monitor or the CRA. This is a professional responsible for overseeing the progress of a clinical trial at a trial site. They provide on-site monitoring of the research study and conduct audits for compliance within the research and regulatory requirements. The CRA or monitor acts as the sponsor's ears and eyes at the trial site, ensuring compliance with the protocol and with the overall regulatory standards. The monitor needs to be thoroughly familiar with the protocol, the investigational product, the trial-specific standard operating procedures or SOPs, as well as the ICH GCP guidelines. They should be considered as an extension of the investigator's study team, but not as police. The monitor is a partner to help the site fulfill all the necessary requirements so that a trial is successful. Working well with a monitor would help a site successfully get through an audit. The CRAs verify that the informed consent is properly obtained, that data is accurately recorded, and that the rights and well-being of the participants are protected. They conduct regular site visits, review essential documents, and provide guidance to site staff to maintain trial quality. And under the ICH-GCP C6 guidelines, CRAs play a key role in risk-based monitoring, whereby they focus on critical data and processes to ensure that there is efficiency and participant safety. In summary, clinical trials are a complex collaboration among many stakeholders, many of whom are seated in this room today. And each role is vital, and the integrity of the research depends on their coordination, compliance, and commitment to the ethical and regulatory standards. Thank you for your attention, and I'm happy to take questions.
Wow. We are grateful to Dr. Freda Mwenda. I hope I got that name very well. For that wonderful presentation on introduction to clinical trial stakeholders. Please, let's put our hands together once again to appreciate her. Well, we are almost rounding off the first session. And this time around, we are going to the next item on this program of event. And it's going to be a question and answer forum. It's going to be presented by one of the research team members of CESTRA and the person of Professor Maxwell Mwebu. And I'm also sure that I murdered this name, right? No, you did it. Thank you so much, yes.
Thank you. Good morning, everyone. And to all the people online, thank you for joining us thus far. So we're going to have a session of questions and answers. But before we start that, let me just say, I know there were some complaints that the audio during the last presentation was a bit, at some point it was lost. Just to let everyone know, including those online, that we're going to share the slides for the presentations. So please just bear with us with that small hitch that we had. So we're going to take questions and answers. The resource persons are still there, those online and those here. Please, those online, please, you can type in your questions and we'll try to extract them. Given that we need to do that extraction, I think we will start with the people who are here physically. So, sorry, it's not like we are discriminating against you guys online, but you will be typing into the chat and it's going to take us some time to extract before we present them. So, the session is open. Questions, clarifications, comments.
Well, good morning, everybody. My name is Mbanefo Francis from Federal Medical Center, Kefi. My own is just a small clarification. When the presenter was talking about the phases of clinical trials, I observed that phase 0 was still with patients. So I wanted to know at what phase do we use the animal model before proceeding into human model, because I saw the phase 0 was still patients, few patients. At what phase do we use the animal model? Thank you.
Thank you very much for that question. I think that's directed to Emeritus Professor Ifeoma Okoye. Please, ma, just note the question. We'll take like 2 more, and then we'll give opportunities for responses. Okay.
Good morning, all. I'm Chinedu Okeke. I also want to ask about the phases. When she was making the presentation, she didn't mention about the OLE, open level extension. So I don't know which of the phase is it going to fall in. Thank you.
Prof, that's another question for you. Which of the phases will the open label extension fall into?
Good morning.
Yes.
My name is Dr. Zoronye. My question goes to the presentation concerning clinical trial stakeholders. In the last slide, she mentioned the CRAs, the monitors. And I wanted to ask, are they independent? Who employs that? She mentioned that they partner with people. Who are they partnering with? The sponsors, the CRO? Who exactly are they partnering to? Are they independently employed to monitor the trials? Thank you.
Okay, so that's for Dr. Frida's presentation. I know she's online, but there's also a very capable hand here to answer the question. So I think at this point we'll just take these 3 before, okay, a 4th question, we'll handle them before subsequent ones.
Okay. Thank you very much for your presentation, Ma. I just want to ask, for the phase 2 of the clinical trials, is there a particular percentage of the patient before they would declare that this drug is not safe for the next phase? Is there a particular percentage that they look for before they declare that a drug is not safe for the next phase? Thank you very much.
Okay. Prof, Emeritus Prof Okoye, that's another question for you. So I think we would go ahead to answer this 4 and then proceed. So Prof, if you can unmute yourself and answer the questions that were directed to you. Prof Okoye.
Yeah, the code was not allowing me to unmute.
Okay. Okay, please go ahead, ma.
Okay. So for phase 0, why are we having an echo? Can most people online, can you mute, please? Okay, that will help us. Phase 0 is conducted in humans. 10 to 15 participants, sometimes patients, sometimes healthy volunteers. And the purpose for that is exploratory, first in human microdosing studies to check early pharmacokinetics, pharmacodynamics, so PK and PD, and target engagement. So the key difference from later phases is that drug doses are subtherapeutic. They are in microdoses. They are not meant to be able to achieve the healing process that the therapy is targeted to, so no expectation of clinical benefit. So in summary, preclinical is before humans. So phase 0 is very early human trial. That's why it's called first in human trial. It's exploratory and it uses micro dosing. Phase 0 is considered clinical and not preclinical. So I believe that that is for the first question. And if we look at, I think the second question was asking about open label extension, OLE study. It's not really its own phase, like phases 1 to 4. Instead, it is an add-on or continuation study that can follow any phase of a trial. And usually it can follow phases 2 and 3, or 3, phases 2 or 3. So look at how it works. An open label extension after a randomized, controlled, often blinded trial ends, participants may be invited to join an OLE study. So in the OLE, all participants receive the investigational drug, and both the researchers and patients, they know it. Hence, open label. It is actually optional and it's designed to collect longer-term safety, tolerability, and efficacy data. So the question is, where does it fit? Most commonly, it fits after phases 2 or phase 3 trials. And sometimes it is used after phase 1 in rare cases, especially in oncology trials. Now, data from OLEs often supports regulatory submissions or post-marketing commitments. So the purpose is to provide ongoing access to the investigational drug, especially important for serious or rare diseases. So that long-term safety and sometimes effectiveness information from OLEs. And you build trust and retention among participants who want continued treatment. So that's what it is. An open label extension is not a separate phase of clinical trials. It's usually a continuation study after phases 2 or 3, and designed to collect extended safety, efficacy data and give participants ongoing access to the treatment. Unfortunately, I didn't get the second. Whilst the second person is answering her question on I think she was asked on the people participating in the trials. Can you then send either in the chat the last question? I didn't get that.
Okay, Prof, I can just reel it out. It says that in the phase 2 trial, is there a number at which point you declare that this particular IP is too dangerous that the safety profile is very poor, and you shouldn't go ahead with it? Is there a number?
Okay.
I think yes.
What usually happens is guided by adverse events. Now, when you have adverse events, you, of course, reporting, and all the different sites are reporting. So when the adverse events are profound, you find that there will be a pause in that clinical trial process in order to do a review and address the concerns. So there is no like a particular number. It's actually determined by the sponsor, because the sponsor is collating all the different adverse events. And I know that sometimes especially during the COVID trials, we were recruiting, the next thing, and we had a target. The next thing, they told us to stop recruiting. They are closing down because the virus has decided to change its nature. Again, another time, we had to end a trial because different sites had targets numbers of participants to recruit. Before we were able to navigate our regulatory issues, the other countries had gotten to the number that the sponsor needed, and they closed out without our contributing to the participants or the information. So these are, closely monitored, and that's what usually happens. Yes.
Thank you very much, Prof. It's good we also have the regulators here. Thank you so much, Prof, for the responses. So I know Dr. Frida is online. If you can unmute yourself and then respond to the question on the clinical trial stakeholders.
Yes, thank you so much, Prof. Maxwell, and good morning, everybody. It's a shame there was a bit of a disturbance during the presentation, but it was lovely to hear everyone's other presentations. So just to take the question on the CRA, so the CRA is the Clinical Research Associate, or the monitor, as we usually call them, and they are usually employed by the contract research organization or directly by the sponsor. So they are not an independent function. And their role really is to monitor the clinical trials, to ensure that it's being done according to the protocol, to ensure that it's being done according to the regulatory and ethics standards. So when we say monitoring, it really means that what they do is that they review the source documents to make sure that everything is captured in a timely manner. They check the informed consents to make sure that all the data has been captured. They do they verify drug accountability. They do training of the site staff as well to make sure that they are well updated on any newer guidelines or on protocol amendments. And also finally, they make sure that any regulatory standards that are already documented, that the site and the site team are following. So that's why we say that they act like the eyes of the sponsor, because they're the ones who are able to make sure that the trial is running in the manner in which it was supposed to.
Thank you very much, Dr. Frida. I think we'll go online now so that they don't feel left out. Please. Are there online questions? I can't see. Please, the presenters, I'm sure you're seeing the questions online. I think we will just go like in bits of fives. So in a clinical trial, who owns the data? Is it the principal investigator, the patient, or the sponsor, or a combination? There's an answer here in the house, is the sponsor. Okay, next one, please. We have this question from Mr. Charles Ubiora. We've not had the presentation on ethics, but let's
Yeah, okay, so that's an interesting question. We've just had a preliminary, a little bit, a teaser. So to answer some of the questions, you have to attend all of the sessions. So this will be answered in the course of the series. You understand? So just ask questions related to today's presentation while the facilitators are online. Thank you.
I get this one is a political question. I think Uwe Modimo Nkata, just to say that research has to be sponsored by someone. So if it may be government, it may be non-governmental, it could be individuals. So basically, the sponsor most of the time funds the research. So if there are a number of researches not being done in most of our health facilities, it's probably because there is no sponsorship. And we know there are challenges. Oftentimes, researchers have to use their own funds to run some studies. So that's a major problem. I don't know how much clinical trials are being sponsored by in-country organizations. So please, next one. Okay, most governments budget for researches. Do we have specially budgeted research funds in Nigeria? And even who manages the funds? Yes, there are government budgeted research schemes. So depending on where you are, at least a prominent one for those who are in academic institutions is the TED Fund. TED Fund supports institution-based research called IBR, and also there's a National Research Fund, an NRF, sponsored by TED Fund, which accommodates larger proposals in terms of funding. Usually the institution-based ones, I think currently the cap is 5 million naira, but the National Research Fund ones is between 40 to 50 million naira. That's a typical example if you're in the academic centers. But there are some other funding organizations. NGOs also do some funding. And then we have quite a number of them. Most of the time, they tend to work with institutions. Most of our health don't have accredited ethics committee. In that situation, who approves the research proposals? Okay. Full disclosure, I'm a member of the ethics committee in our hospital. So I will tell you that I don't know if the NREC rep is here. Okay, he hasn't, okay, he's online. Oh, okay. Mr. Ado, if you're online, thank you for coming. Now, basically, when they give ethics committees licenses, there are grades, and depending on your grade, your coverage is accommodated by that grade. So if you're in a facility where you do not have a committee and you need to do your research, you can seek ethical approval from contiguous organizations or facilities where you have proper recognized ethical committees. Next one, please. This sounds like NAFDAC question. I don't know. Dr. Beno, would you want to help?
Okay, maybe to go through these questions. Again, a placebo should come alongside with the product. So you cannot just get placebo without having the IP generally. So I don't expect you to have placebo without you having the active substance you're going to do the study with. So I think that's what it is.
Yes, actually, the sponsor provides it. If there's going to be a placebo, it comes with the products, the IP and the placebo. They all come together. And they are all included in the protocol as well. And I saw on the chart, somebody was asking about the fact that I talked about primary endpoint. There are still some people unmuted, so it's echoing. I don't know if you can mute. So for secondary endpoints, the additional outcomes that are measured to provide more information about the intervention. And usually the purpose is to explore other potential benefits or risks and to support understanding of the drug's profile. So the characteristics, for instance, is still pre-specified, but not the main driver of the trial design. And it can include safety, tolerability, quality of life, or exploratory efficacy measures. And they are useful for hypothesis generation and supporting evidence. And the examples are like in the cancer trial, for instance, tumor response rate, the quality of life scores, and biomarker changes. And then for another example is in a hypertension trial, diastolic blood pressure, a proportion of patients achieve the target blood pressure. And in the vaccine trial, severity of illness among breakthrough cases and immune response, antibody titers. So a simple analogy that you can use is that the primary endpoint is the main exam grade and decides if you pass the course. And the secondary endpoints are class participation, assignments, projects, help show you overall performance, but don't replace the main results. So that's an analogy. So the summary, primary endpoint, the single most important question the trial must answer. Then the secondary endpoints is the additional outcomes that provide supporting evidence.
So there was a question.
Thank you.
Yes, thank you very much, Prof. Thank you so much for your words of wisdom. There was a question about why clinical trials are not commonplace here. And this is why this is a capacity building workshop. It's 12 weeks, and we want you to make a commitment to go through the process and to get the certification. There is something that investigators can do to make themselves trial ready, and that is to go through all the GCP training and health ethics training. These are mandatory requirements before anybody will even look at you, apart from the fact that you're qualified. Then there are some things that your institution must do, certain equipment and facilities that they have to have in place, making sure that everything that you're using to look after patients, all the equipment are being calibrated. These are some of the things. And then as an investigator, having a registry, a record of all the patients that you're seeing is very important. So if you're a clinician and you're working, you will have a registry of the patients. Begin to have that registry. That's one thing that you find very useful, because when it comes to feasibility, you'll be able to go through the registry and pick out the relevant participants but one thing is training. And you're not just going to get trained once. Like if you look at the GCP, it's every 2 years you're reminded, the training, needs to be refreshed. So these are some of the things that we need to do in order to make ourselves ready. So recognizing the importance of clinical trial, IQVIA has sponsored this training, and I hope that all of us will take advantage of it. And it will be one in a series of many such trainings. Thank you.
Thank you very much, Prof. I think, I don't know, maybe we'll take 3 more questions before the break. Any house? Oh, okay. Come to the high table.
Thank you. My question is directed to Kathy Grab. I don't know if she's still online. I'm happy to note the robust partnership they have in Nigeria. But I'm interested to find out if how do other CSO or hospitals partners get involved in the hospital partnership that they are partnering with. So if a hospital is interested, how does that hospital apply to become a partner? Thank you.
I don't know if Kathy is online. Can you people see her? Is Kathy Grave, are you online? Because what she sent in was a recording. And I think time difference because she was coming in, she's based in the US, Washington. I'm not sure if she's online. Would address that to her, and in the course of this, we'll send the response to you. Any other in-house before we quickly just go to the online questions? Please, can we see the online questions? So we just, okay. Dr. Ekwere is one of the SparkNet site leads from Akwa Ibom, Uyo. And he's asking, why do some clinical trials fail despite promising laboratory results? I don't know who I'll direct this to. I'll start, Emeritus Prof.
Okay. Yeah. Okay. I can take a look at that. Why some clinical trials fail despite promising laboratory results. You'll be looking at issues of human biology. Human biology is quite complex. Then you look at issues of dosing differences, safety concerns, trial design issues, and civil or real-world factors. I believe that I think I saw in the agenda, but let me just quickly look at it. For instance, the human biology is more complex. What works in a petri dish or an animal model often does not translate to humans. Diseases in humans are influenced by genetics, environment, and lifestyle factors that lab models cannot fully capture. And then for dosing differences, the dose that looks effective in the lab may be unsafe, may be toxic, or ineffective in humans. So finding the right balance between efficacy and safety is often where many drugs fail. Then in terms of safety concerns, you'll be looking at side effects may not show up in animal studies, but can appear in humans. And some toxicities actually may emerge when drugs are tested in larger groups over longer periods. And hence, you can imagine, the vigilance that is needed. Then trial design issues, poorly chosen endpoints, inadequate patient selection, or small sample sizes can make a promising drug look ineffective. And sometimes the wrong population is tested, meaning the real benefit is actually hidden. And placebo and real-world factors in humans, the placebo effect can actually reduce the apparent difference between the drug and the control. And variations in patient adherence, comorbidities that exist, or lifestyle factors also influence the outcomes. Sometimes regulatory and ethical standards, even if the lab results are positive, if the trial doesn't meet GCP or ethical standards, it may still be stopped. So these are some of the reasons. that humans are far more complex, and a drug may not be safe at effective doses, may not work in real patients. Thank you.
Thank you very much, Prof. I don't know, is there any other online question? I think I can.
Please, they are fully involved in nurses, pharmacists, and medical lab scientists. In our own team, we have, you can't do without all these team members, medical lab scientists, the pharmacists, the nurse. They are all stakeholders. I don't know where it was not mentioned. Remember what Professor Obi has just told us. These are just like introductions, a kind of overview. And then as you go along, you are going to see more.
Thank you, Prof, for that. I also want to say that even for this workshop, the SparkNet sites, amongst the key people that were requested to be part of the team that should, at each site, participate in the 12-week workshop, we also had the laboratory people there. So thank you for that. Then I think one more question. I said we'll take 3 online, and then we'll look at our time. Please, do we have any clinical trial research group that is coming up? Because up till, we're still collecting full of data for SparkNet. Prof. Professor
If we have any clinical trial research that is coming up.
Yes, thank you very much. So this is why we are doing this. We have a registry. We'll get ourselves ready. We make sure we are following up our patients regularly. We have access to the patients in the registry. And then when the opportunity comes, it is the sponsor that does feasibility. The reason why we're having this training is to make sure that all these SparkNet sites can qualify to be part of a clinical trial. And they will come independently to you as an investigator in your health facility and see if your facility qualifies. So that's why we are building capacity across board, because we don't want to be the only place, or maybe Lut or Ibadan, Enugu, that are conducting clinical trials in sickle cell disease, or Kano. So we want everyone to be able to participate. So please just get on board, do what you're supposed to do, capacitate yourself, make sure that your institution is providing the facilities that you require. And then when the sponsor sees that you're ready, they may invite you to be part of a global study or a national study. Thank you.
Prof, can I add to what you've just said? I don't know if I'm permitted to add to what you just said.
Of course.
Okay. There's what is called Clinical Trial Readiness Index. And, at first, when you come up with clinical trials, everybody's thinking a lot of infrastructural, equipment and all that. Let me tell you, the main, like to call it equipment, is the workforce for clinical trials. It is the human capacity. It is why as soon as you get the human capacity right, the GCP will sit in place. And then every other thing will. You find that most of the equipment you have in most hospitals will suffice for most of what is needed for most trials. So we're not looking at, humongous amounts of laboratory equipment, infrastructure, but we are looking at human capital. And that is what we are here to develop. Thank you.
Thank you. Sometimes they want you to have thermometers that will be able to give an alarm, right? To alert you at home if there is a deviation temperature. Thank you.
Thank you, everyone. I know there are still some other questions, but we need to move on to the next segment. So we'll note those questions, and in the course of today's program, we'll try to address them online. So over to you, Dr. Hadi.
Please, let's have a round of applause. Yes, it's been quite an engaging question and answer series. Many of you have asked very probing questions, and I'm quite sure that you've got very profound responses from our resource persons. As I said earlier, we're almost rounding off the first segment of this program, and you can see many of our distinguished members of the press are here. The next session will be for them, which is a press briefing, and we shall be inviting to the high table our distinguished resource persons and erudite scholars who will be providing some answers to some of the questions they will be asking. Of course, basically, this is going to be only for the media. You have had your session, you've asked very interesting questions. So this time around, it's only for the members of the press. But before we go further, my attention has just been drawn to the fact that there was one of us who was not introduced. On this note, I would like to introduce one of the members of IQVIA, right? Dr. Olaoluwa Akinlolua. And maybe this is a time for me to also introduce my humble self. My name is Dr. Habib Yakub. I'm the Acting Director of Information and University Relations of this great university. A round of applause for me. On this note, I'll be calling to the high table 4 of our erudite scholars, as I said earlier. Let me invite Professor Obiageli Nodu to the high table. Please, a round of applause for her. I will also be inviting Dr. Kweku Marfo and the representative of the DG of NAFDAC, Dr. Beno Yakubu. And of course, the professor has just left here, Professor Maxwell Nwebu, right? Well, and as you rightly know, we earlier introduced all of these personalities. This is another forum, and this is the segment that we round off this first segment before we move into the second one. Let me reiterate that this is basically for the members of the press. We shall have a brief response or comment from any of them or all of them before the floor is thrown open to the members of the press to engage them. Once again, please, let's have a round of applause for them. Okay. Professor, the floor is yours.
Good morning, everyone. Good morning, ladies and gentlemen of the press. It's a pleasure launch of a 12-week clinical trial capacity building workshop, which will take place from the 18th of September to the 27th of November. It's an intensive course which we would want everyone who actually wants to benefit from the training to participate fully, because you can see that every aspect of the course has been carefully packaged to be able to meet the skills gap that we have in this area. You can see what one small session this morning has unfolded to all of us, and that is how all of the sessions have been prepared to deliver to the participants over this period. We at the National Center of Excellence for Sickle Cell Disease Research and Training at the University of Abuja are quite excited to partner with the largest clinical research organization in Africa, sponsored by them, to bring this innovative training to stakeholders within our community. And if you watch online, there are people not only in Nigeria, but across Africa. So this is exciting, and we have launched to have this capacity building. And we're very grateful to have IQVIA and BioVentures for Global Health, help us to get to this point. So why is this training necessary? Like I said earlier, it's not just enough to have a disease of high burden, a genetic blood disorder like sickle cell disease ravaging the people on this continent from infancy, to middle age, if they can get to that. But it's also important that we leverage the high prevalence of that disease to be able to bring solutions. And we have often said that if you're able to manage sickle cell disease, have facilities to manage it, then you can also do for almost every other disease, because it is a disease that has complications, lifelong complications from infancy, to adulthood. And these complications involve almost every organ system in the body. So what has happened is that over the past 8 years, we have been able to benefit from an infrastructure grant from the NIH as part of the Sickle Pan-African Research Consortium to build a registry of almost 40,000 patients across 6 countries in Africa, out of which Nigeria has contributed over 10,500 patients. And we are prospectively following up these patients in the registry. So it's not enough to follow up the patient. What are we offering to these patients? We are now having innovative treatments, novel treatments for sickle cell disease. And we even have some not so new ones like hydroxyurea, but which is not being used as as commonly as we ought to. So we want to make sure that all the physicians, all the healthcare workers who are looking after these patients with sickle cell disease are prepared to participate in the trial of these novel treatments. So that is why we having come out of that 8 years experience, we want to make sure that all the sites are trained, and they can participate in these new therapies that are coming up. Because, like I said, Nigeria should not just be bear the global burden. We should bring the solution, help to bring the solution to the problem of sickle cell disease in the world. The other point is that what we are doing is in keeping with health policies within the region, both by WHO and also by our national authorities. And we are particularly delighted to have NAFDAC here, because somebody once said that sometimes before even when we are approved, before we get to the actual point of participation, there could be some delays. So we are happy that NAFDAC has been working with us to get us ready, and they've been building the ecosystem over many years to make sure that all the regulatory processes are in place within the country to help investigators and to help institutions to participate in clinical trial. The other thing is that this is Sickle Cell Awareness Month. September globally is the Sickle Cell Awareness Month. So this is part of our activity, for sickle cell disease awareness in 2025. So we want to work across 25 sites in the country, and then of course, other sites who may not be partner sites to work together to develop this capacity to launch us into the next phase. So the mode of delivery of this course is weeks 1 and 2 is being done, in person at this workshop, and then we have 10 more weeks, which will be online every Thursday, 7:30 p.m. for 1.5 hours, and we are going to have a really interesting time. If you look at the curriculum, and they have been placed in the jotter, page 3 of the jotter is the program. And you can follow, you can see that is every aspect of a clinical trial, it has been covered by this, and then we'll be putting resources online, for us to follow. All of the lectures will be warehoused at the center, afterwards, so that people can go online. But every session is mandatory before you can get the certificate, just to reiterate that. So the expected outcome is that, like I said earlier, at the end of this, would be quite prepared in terms of understanding the fundamental concept of clinical research and trials. We'll be able to differentiate between the different phases of clinical trials. We know that clinical trials takes place under certain ethical and regulatory framework, so we understand what that is, and then we'll be able to describe the role of stakeholders. We know that stakeholders in health, stakeholders such as you, the press, are also important, because sometimes we have to come to the press to help us to advertise in terms of sending the message out in order to recruit an appropriate number of patients. And then when we understand the ecosystem, the way trials are designed and run, then we'll be better prepared to participate in the process. So we are very grateful to IQVIA for sponsoring this, and then for is a global leader in this space. We are very grateful to have them. We are also grateful to have BioVentures for Global Health, get us some participants. They actually laid the foundation for what we are doing 3 years ago. For us, specifically as CESTA. But when we wanted to expand and upscale, then we called them again, and then we are able to have this. So one of the exciting things that we want to do at the end of this is we have in mind a paper. Most of us know that there's a paper which is very well cited. It's called Current Management Practices for Sickle Cell Disease in Nigeria. That paper is very well cited. And all we did in that paper was just to describe what we are doing in terms of management of sickle cell disease. At the end of this, we want to have a paper that is describing the gaps and challenges of conducting clinical trials in resource-poor regions, the practical experiences from ongoing clinical trials at CESTA and other partner sites. We predict that this will be a well sought after paper by sponsors and everyone in the space, even as we write it. So I want to encourage my colleagues, who are PIs, to respond, quick as you have always done, very commendable, to respond so that we can shape this paper in collaboration with our sponsors and our regulatory agencies to be able to deliver that. So thank you very much.
Okay, thank you, Prof, for this wonderful speech. Again, on behalf of DG NAFDAC, who I'm representing, I want to thank the organizers and sponsors of this program for bringing NAFDAC into the fold. Again, NAFDAC as a regulator, the main function of the agency is to make sure that all the drugs that are used in this country are of good quality, safe and efficacious. So again, our role in regulating conduct of clinical trials in Nigeria, it's very, very crucial. We want to see all clinical trials that come to this country meet the best standard globally. And again, based on that, I think NAFDAC has undergone global benchmarking through WHO. And I'm proud to announce that with the effort of the DG, we scaled through maturity level 3, again, which very few African regulators have gone through that stage. And again, that means that it translates to any product that is approved in Nigeria is trusted globally. And at the moment, she's working towards becoming WHO-listed authority, which will mean that we can develop even vaccines from the scratch from Nigeria and approve and use globally. So that being said, we are delighted that we are part of this, meeting our stakeholders. And again, as an agency, we are committed to improvement. If there's any area that we might not be doing well, I think we welcome our stakeholders to punch us. We will improve. Because as we all know, the biggest room on earth is the room of improvement. We will continue to improve till one day we become like USFDA. And that being said, I want to thank you again for bringing us in. And my DG is well delighted that we are part of this success. Thank you.
And on behalf of IQVIA, I'd like to recognize Prof. Nodu and Sestra for the opportunity for us to sponsor this event. IQVIA, as a clinical research organization, we've been operating on the continent for over 30 years. Most of our staff are based in Africa, and we really support institutions like University of Abuja Teaching Hospital, Sestra, as well as other institutions on the continent to really play a pivotal role in bringing innovation to patients. As indicated by my team, sickle cell disease really have a significant prevalence in sub-Saharan Africa. We are seeing emerging innovative therapies coming to market, including potential curative therapies like gene therapy, and certainly building capacity and engaging with stakeholders like yourself to ensure that we could play a pivotal role in bringing these innovations to patients will be critical. So on behalf of IQVIA, we appreciate the opportunity to partner, and we're looking for more engagements with you all across the ecosystem. Thank you.
Thank you very much to everyone. I think mine is just to say a big thank you to everyone who's helped to see the actualization of this. I want to particularly thank IQVIA and BVGH for the faculty that they've provided to us this and the support that IQVIA has given us for us to be able to engage in this process. And also to thank the regulatory bodies, NAFDAC and NREC especially, not just because of those workshop, but also for previous engagements with them. I think the progress we've made at CESTA in the past couple of years with the number of trials we are doing, we wouldn't have been able to do that without the support. And I know the director of CESTA had mentioned it before that we successfully went through a NAFDAC audit process, and they really supported us and, of course, opened our eyes to various areas that we needed to have improvements and which we have implemented. And to say to everyone, including those online, you're starting now similar to how we started some years back. It looked like when they talk about clinical trials, it was this big thing, how are you going to go through it? But I remember when Professor Lopade and the team came from University of Chicago to train us, we finished the training and we went on under the BVGH clinical trial training, and here we are today. So it starts just like this. And I want to encourage everyone who has registered to please ensure that you go through the 12 weeks. Certification, like has been mentioned, will only come with mandatory attendance of all the sessions, and as most trainings are in clinical trials, passing to a certain level, a certain percentage pass at the end of the training during the assessment period. So once again, I want to encourage everybody, and I hope we all have a good training going forward. Thank you.
I just want to ask, what is the role of regulatory agencies like NAFDAC in ensuring that, the process is fast-tracked from investigations to clinical trials?
So the role of NAFDAC as a regulatory authority is to make sure that for every submission of clinical trials, we weigh the risks, benefit risk ratio of this study design, as well as to make sure that the protocol is good enough to be implemented and is in line with our regulatory requirement as well as global best practice. So if you have all this information in your protocol, in your submission, we don't have any reason not to grant your approval. However, we have a turnaround timeline of 60 work days to give an approval. But most of the times, I think if we have a less complex study design and the applicant meets all the regulatory requirements, it doesn't take up to that 60 work days. But again, we have another pathway called fast track pathway, whereby we depending on the particularly during emergencies, we take far minimum less time than the normal approval timeline, which could be 10 or 15 work days. Again, depending on the particular product you are studying. For a novel product, during emergencies, it takes us 15 work days. Then for a repurposed product, I mean product that but it's being repurposed for another indication, it takes us do that. Thank you.
Okay. Yeah, good morning all. My name is Felix Kanova from Authority Newspaper. So my question is to Prof Nodu. I want to know the number of participants taking part in this training course. And will there be examination at the end of the program to assess, to know whether they have been able to achieve what is expected? Thank you.
Okay. Yeah. Thank you very much. I must say that this appears to be a course that has drawn a lot of attention. We designed it for SparkNet sites. There are 25 clinical sites within the network, and we have mandatorily 5 persons from each of those 25 sites. But we also know that these are the ones that will be directly supported. But every PI is encouraged to offer the training, because it is online, to other people. Like when you saw the number of persons who are involved in delivering comprehensive care, definitely more than 5. So I would expect that probably the minimum in each site will be 10 who will go through, but there can be more. We've had Professor Maxwell whisper to me this morning that we had about 460 registrations online. And when we put this on LinkedIn, we had over 16,000 views, showing that people, and some engagements, showing that people are interested. It's virtual, so that has the capacity to increase the number of participants as they become aware of it. And then, of course, when we finish, it will be warehoused on our website, and there will be a mechanism for letting people also participate, go through the course completely. Thank you. I forgot to say that there are also people outside of Nigeria who are like Sudan, Zambia, and other countries within a larger consortium that are participating. Thank you.
Okay, good morning all. My name is Fred. I work with the Sun newspapers. I just want to have an information on this trial, which is focused on sickle cell diseases. I don't know if you're discussing maybe if you can tell us possibly which research you're doing on this sickle cell disease and the new inventions or maybe new method of curing the disease or finding a new solution to it. And if you're doing that, what is the target timeline for possibly developing a new way of attending to this disease, which is currently, like you said, Nigeria is one of the highest burden in the world. Thank you.
Thank you very much. Just to reiterate that this is a capacity building course to build the capacity of healthcare workers to participate in clinical trials. We are not targeting any specific treatment. We are building the capacity of sites to participate in future clinical trials in sickle cell disease. And like you heard, if you train to do clinical trial in sickle cell disease, you can also apply the same training to other areas like in oncology, in hypertension, in diabetes, in pulmonary disease. So it's a capacity building course in clinical trials. Thank you.
Well, thank you so much for your questions. Well, the distinguished members of the press have told me that they have had enough, and they've interacted among themselves, and they have an angle they probably would want to be looking at their stories. Well, let me use this opportunity. I think, okay, well, there's refreshment, which is an important announcement, but I was going to come to that, yes. But it's important that it comes first, so that whatever I'm going to say, at least this is in your head, and you know that you have a refreshment. So I think with this press briefing, we are going to draw the curtain on the first segment of this program. As you rightly know, this is just the first segment. You have a lot, and we thank you sincerely for finding time out from your very busy schedule to be here to attend this program. And we appeal to you and urge you very strongly that you shouldn't go away so that you can attend the next phase of this program. Of course, I've been told that they are very, very important for you, because if you just attend this one and leave the other one, it's as if you haven't come at all. Once again, I thank you so much for coming, and let's have a befitting round of applause for ourselves. Thank you. Press, yes. Go first. Prof said you should go first. Thank you.
In NAFDAC, Dr. Benu, to come and take us through clinical trial regulatory guidelines and processes in Nigeria. Please, let's welcome him.
Thank you, sir. Once again, good day to everyone. And just like I've been introduced, my name is Ben Nyam Yakubu, and I work for NAFDAC as the head of clinical trials in NAFDAC. So my presentation today is to talk about the guidelines that drive our processes. Again, I think the presentation made by Professor Okoye really softened the ground. Again, that will really give us more understanding when I present these slides as regard to what we do as an agency. And just like I've stated, I think one of our main mandate is to make sure that the participants' well-being are well protected, safeguarded, as well as the credibility of the data that will come out from those clinical trials. So I think that being said, I want to continue. So, okay, this is my outline. I will talk about introduction, definition of some terms, then the roles of a regulator, NAFDAC particularly, as we are talking about it now. Then we talked about the enabling laws in regulation of clinical trials. Then we talk about the roles of determining scientific suitability of clinical trials applications. We talk about assessment of safety of an ongoing clinical trials. Then we talk about continual review of a longer term clinical trials. We receiving an assessment of end of study reports, then conduct of GCP inspections. Again, why I put it end to end is because our guidelines that I'll show you comprises everything that we need to know so that when you're applying for clinical trials from the beginning to the end, you have at least a brief knowledge about what NAFDA does. Okay, so in the introduction we talk about telling you that clinical trials are a key research tool for advancing medical knowledge and patient care. Very important in discovering new treatment, just like Ifoma made mention today. Again, without it, we're just doing blind treatment. So it helps us as well to have new treatments, as well as new ways to detect. Oh, okay. Sorry, please. Hello? Better now? Okay. Okay, I'm sorry for that. Again, let's move on. And as I was saying, talking about introduction, why we need to conduct clinical trials, this will enable us to be able to discover new treatments, new diagnoses, as well as a chance of developing new preventive measures for diseases. Again, so we are a government body, that's regulators are government bodies responsible for effective drug regulation as required by the law. And this will enable us to be able to make sure that the outcome, the product is safe, efficacious, and of good quality for use by our people. So that is why regulators are there to protect the lives of the populace. Can somebody help me on this, please? Okay. So the definition of a clinical trial. So Prof did, but I think let me just, based on our guidelines, which is not quite different anyway. They say any investigation in human subjects intended to discover or verify the clinical, pharmacological, and or other pharmacodynamics effect of an investigational product and or to identify any adverse events to an investigational product and or to study absorption, distribution, metabolism, and excretion of an investigational product with the effect of ascertaining its safety and efficacy. Again, if you look at what we do, maybe later in the process you'll see some of the scope of our activities, which include what we call bioequivalence. Again, the definition encompasses everything, but the core clinical trials and the bioequivalence, that is to be able to ascertain that generic product is as good as the innovator product. So, the general role of regulatory authority, please can you help me put this one up? Okay, I should use this. So the general role of regulatory authority as NAFDAC is we play a very crucial role in making sure that good clinical practice in clinical trials regulation across the world is done in accordance with the best global practice. So what we do as a regulator, we prepare and maintain regulation by establishing and updating regulations that govern the development, manufacturing, testing, and marketing of pharmaceutical products, which the initial concept of is to approve it, ascertain that the product is good to go to the market. After the development, then you start the process of manufacturing. You now pull out the quality data, putting it together into a dossier, product goes for registration. Then as regulators, we regulate the conduct of clinical trials to ensure ethical conduct and the scientific record of all these studies. So we also evaluate clinical data submitted by pharmaceutical companies to decide whether to approve the product or not. And it is only granted approval when we ascertain that the benefit outweighs the risks for our population before we can do that. Without that, it will be difficult for us to approve clinical trials. Can you go to the next slide, please? So, again, one of the roles of regulatory bodies is to critically review preclinical and clinical data to assess safety, efficacy, and quality of data submitted by pharmaceutical companies. Again, for us to begin any review of any submitted clinical trials, we look at the previous data. And again, Prof talked about the preclinical that is done maybe from the lab to the animal model, down to human beings. So we make sure we review this data when it comes to us to ascertain why it should be extrapolated to humans. Make sure that it's safe and shows a level of, if it's a vaccine, maybe immunogenicity, if it's a drug, shows a level of safety in animals before we can be able to allow it to be used in humans. So at that stage, we require you submit all those information to us to review. So we also have the responsibility to close down any ongoing trials in the case if there's any serious breaches of good clinical practice guidelines. Again, we have executed that mandate sometimes when we realize that a study was ongoing and unfortunately the comparator product was already expired, again, which was a high risk to the people who were being used for that clinical trials. So we have the mandate to make sure that all the information on the product label has been determined. And these are determined through clinical trials because all those adverse events, all those safety precautions, they are picked during the conduct of clinical trials. So regulators make sure all those things are reflected when the product has eventually gone back, want to go for marketing authorization. So we also set standards for drug quality to ensure that drugs meet certain quality requirements that our people will use. Again, we work together with other regulatory authorities in form of reliance to make sure we have good products home to Nigeria. For instance, we could have a product that is coming in and has been reviewed or has been approved by another regulatory authority that we trust so much and we've signed MOU with. We can rely on their review records and allow this product to be registered in Nigeria. Though we will have the review reports, then we'll review the review reports, then we'll move on to approve this product. Next slide, please. Then, for post-marketing surveillance, we monitor drug safety and effectiveness post-approval by collecting and analyzing data on the adverse reaction and other safety concerns reported. Again, we can do that because we know that sometimes when you do a control study and the product goes out to the general public, you don't know what is going to happen. So as it goes to the uncontrolled population, you could discover some adverse reactions that might happen. Remember the case of sometimes, I think, GSK at the time they first developed their I think, rotavirus vaccine. Again, they picked few cases during the study, the clinical trials to the point of approval. But again, when it got out to the market, they discovered the cases were more frequent to the point that it led them to the regulatory authority had to withdraw that product in the market. And they had to go back to the drawing board to develop what they have today. So, as for us, again, we conduct good clinical practice inspections, both for clinical trials, we conduct for manufacturing facilities who produce those investigational products, because we must make sure that the product produced for those investigational purposes met the good manufacturing practice to make sure the quality is guaranteed. Again, you could maybe say that at the point of maybe early phases of the studies, yes, at that moment you haven't developed the product, the quality attributes of the product, but you are expected to produce this product in a GMP certified facility. So we demand for that as you submit application, both for clinical as well as for investigational product permit, import permit. Again, we do manage pharmacovigilance system to track and evaluate adverse drug reaction and other safety-related data addressing potential safety issues associated with drugs. Again, for us, there could be some data that may come, maybe we know that our population need this product. We could, based on the initial report, allow this product to go to the market, but we keep going on to make sure that the applicant or the market holder conduct other studies. We could do post-approval safety studies. We could do post-approval efficacy studies. Particularly for vaccine products, most of the effectiveness is done after the product has shown evidence of efficacy. So at that point, it's used in larger population. You can do that bridging study to get what you want. At least that will really optimize your product quality in the market. Then we play a role in educating the public about the safety of medicine and device use. Again, sometimes we do organize some outreaches. We do sensitization. Mostly in this country, we collaborate with NREC because they too, they are a regulatory authority in the aspect of ethics to make sure we enlighten our people, particularly when we have new information or new guidelines in place. We just did one in March when we have this new ICH guidelines, E6R3. We had that workshop in Lagos, and we did that for bioequivalence because we've just developed a new policy to make sure we regulate bioequivalence for generic products that qualify for it in Nigeria. Next slide, please. Right. So, what gives us the mandate to do all these things? Again, we have a law that mandates NAFDAC to regulate the conduct of clinical trials and to make sure all studies are conducted in accordance with the national and international guidelines for good clinical practice. Next slide. And these laws, we have what they call NAFDAC Act Cap 1, Laws of the Federation of Nigeria 2004. This law gives us the mandate to regulate and control the manufacture, importation, exportation, distribution, advertisement, sales, and use of food, drug, cosmetics, medical devices, packaged water, and chemicals. All these products are called regulated products. So if we say regulated products in NAFDAC, it means all these products. Again, with this overarching law that gives us the mandate to do what we are doing, now the parliament decided to put particular laws for the regulation of conduct of clinical trials in Nigeria, which we call it the Food and Drug and Related Products Registration Act, Cap F33. Again, Section 5.1 and 2 empowers NAFDAC to regulate and control clinical trials in Nigeria. And this section specifically stated that no person shall, in the course of his business, import or supply, procure the importation or supply, those are the legal English anyway, or procure the manufacture or assembly of a drug product, cosmetic or medical device for the purpose of clinical tests, unless he is a holder of a valid clinical trial certificate. And for us, that is the approval. And the trial is to be carried out in accordance with the terms of the certificate and the provision of any regulation in force. Again, this is the act that gave us, expanded the mandate to control the conduct of clinical trials in Nigeria. And arising from this act, we now in-house develop our guidelines that is enforceable. You can enforce these laws as well as the regulations. Next slide, please. So, the scope of our activities in Nigeria, we requires clinical trial to be conducted on new or relatively new chemical entities. Again, when we say new, a product that has started its conception in Nigeria, it's a new chemical entity. Again, what we call relatively new, in quote, products that are coming to this country maybe hasn't been here before, and when you look at the data submitted to us, it might not have any data containing that was, the study was done in a population similar to ours. Again, we know that there's what they call pharmacogenetics. So without the data, we need to do what they call a bridging study here. You extrapolate what you have gotten there to replicate what you got there from our few population here. So that will give us that confidence that yes, the data you have there is to be reflected here on our population. Then we'll be comfortable with that and we'll move forward with it. Then, for drugs of new indications, yes, you could have a paracetamol today and begin to tell us that paracetamol too can cure maybe cancer. That's a new indication. You need to demonstrate that through the conduct of clinical trials. Then we talk about, we do on new patient population. Again, if you have a product that you've been indicated only on adults and you want to extrapolate it to children, just like what happened during the Trovan. What really happened in Trovan case was that Trovan was already a registered product in the US for adult population 18 years and above. But again, when there was a problem here, the Pfizer brought it in the guise of assisting our intervening during the emergency, during the outbreak. But again, in this guise, they tried to use it in children to see whether they will get the same thing. But again, they did not follow the proper channel of conducting clinical trials. That is why they had that issue. Then new combination drug product, if you are bringing two entities together, you know you've tampered with the safety profile. It could be either synergistic or the other one can cancel the other one. So we need to demonstrate that when you put this combination together, they're going to give you advantage over individual moieties. So you need to demonstrate that through clinical trials. Again, you have new dosage regimen for which safety and efficacy profile have not been determined. You need to determine that through clinical trials. Then, if you have new delivery or medical devices, again, you need to prove that it can perform what it's designed for. Then, now we are talking about bioequivalence and bioavailability study for products, injectable, and tablets who qualify for bioequivalence. And the purpose of this is that, remember, 80% of our drugs in Nigeria are generics. So again, sometimes we've had issue whereby people complain that they took this drug and there was no effect, therapeutic effect. So we want you to compare your generic product with the innovative product to make sure that therapeutically they are equivalent. It might not be the same, but there's a range that they must cross to be able to be acceptable that, yes, it's as good as what you have in the market. So the instrument we use to regulate clinical trials in Nigeria, we use regulations, we use guidelines, and internally we use standard operating procedures. Again, we had been using a draft regulation before 2021, but we got our draft regulation gazetted in 2021. We've been falling back to our parent law to regulate clinical trials, but in 2021 we had our regulation gazetted, and this is to ensure that clinical trials in Nigeria are well designed, conducted in accordance with national and international ethical and scientific standards, using the good clinical practice principle and the Declaration of Helsinki for ethics. Then it further explained circumstances in which clinical trials shall be considered, as well as penalties and administrative sanctions for non-compliance. Again, I'm sorry to say, I think somebody was asking today whether we do have some studies that are not in accordance with the law. Definitely we do have them. And I believe even here, you may have some clinical trials that are ongoing without passing through regulatory authorities. Again, it's against the law. And again, when it's discovered, it can be sanctioned. The PI can be sanctioned, because there's no reason why you should expose people without getting your process or your protocol approved, both by regulatory authority as well as ethics committee. So the guidelines are available. The available guidelines are documents for the general public, and in particular interested parties that may want to engage in clinical trials of NAFDAC regulated products in Nigeria. And the guidelines help to expand the requirements of the regulation, but are non-binding on the applicant. They are not enforceable. We enforce the laws and the NAFDAC Act. So then we have, like I said, we have our SOPs that are for internal use. It guides us in what we do. Next slide, please. So part of the instruments are the guidelines we are talking about. So we do have guidelines that guide our processes. These guidelines to guide our stakeholders for easy compliance. So we expect that before you come for seeking approval for clinical trials, you might have gone through our guidelines, get yourself familiar with our guidelines, and again, if you have any doubt, we have a provision in NAFDAC that you can activate. We call it pre-submission meeting, which is free. You could just send an email telling us you want to do a pre-submission meeting. And the essence of this pre-submission meeting is for us to get to know what you want. You get to know what you want. If you are not too familiar with our guidelines, you ask all the questions in the world for you to get clarification from us. This will really help to minimize the back and forth when you submit an application. And again, we have another system called scientific advice. If you so wish, if you want to start a study, you want to maybe help us to guide you, look at your design, look at your processes, you can activate that, and we look at it, guide you scientifically on how you go about doing it. So we do have guidelines for clinical trial application in Nigeria, which contain all the necessary documentation you need to present to NAFDAC for us to review and grant you approval to conduct clinical trials. Then we have guidelines for clinical trial protocol development. Again, for those who have never done it, these guidelines will really help you, because all the nitty-gritty of a protocol, the components of a protocol is there, and they are self-explanatory. If you go through that, you can at least design 50% of your protocol, then you seek assistance from other people. So that guidelines is up there on our website. Then we have guidelines for good clinical practice. Again, we adapted it from ICH guidelines E6 R3. That's why it's 2025. So it's on the website as well. We have guidelines for labeling of investigational medicinal products, because the labeling for the IP are quite different from labeling of products for registration, because there are some certain information you must put on your labels for IPs, not as you're going to get in products meant for registration. Then we have guidelines for importation and release of investigational medicinal product. There are certain criteria of products and the way you apply and get it down to Nigeria. And get it released if it's biologics or vaccines. So guidelines for submission of clinical trial progress report. Again, we expect that when your approval is given to you and you start implementing your protocol, you should be giving us progress report quarterly. That will really help us to track what is happening at the site, even if we don't come for GCP inspections. Again, guidelines for clinical trial process timelines. These guidelines stipulate timelines for every activity we do. Again, just like I said, 60 days for CTA reviews, 15 days for expedited review, and all those things. The guidelines contain all those timelines. It will guide you when you're about to submit your application. Then we have guidelines for trials in pediatric population. Again, knowing before now, in fact, the Global Society has been saying it is not good to conduct clinical trials on children. But again, the same Global Society scientific community look at it and say, look, all the drugs we've been given to children has been extrapolated. Why can't we just try do studies that are for diseases that are particular to children? That is why we've quickly drafted guidelines and it's enforced. Then we have guidelines for conduct of clinical trials during emergencies. So we activated this one during Ebola as well as during COVID-19, and it's enforced as well. Next slide. So these are our processes that are driven by these guidelines. We have, we do receive and review clinical trial application package. We do receive and review application for IP importation, that's investigational product importation. We do review clinical trial safety reports as it comes to us. Next slide. Then we review clinical trial progress and final reports. You give us progress when the study is ongoing. After the study is completed, you have timelines. You have to submit your final report. We review clinical trial protocol amendments. You could start a study and there's need for you to amend your protocol. You need to get approval, particularly if this amendment is substantial. You need to submit it to us to get approval. Back, please. Then, for the conduct of clinical practice, GCP inspection, which is very crucial, is one of our pivotal role we play because, again, it's not enough for you to approve a protocol to be implemented. Most of these non-conformances are discovered during inspection. You will see a very beautiful protocol, but going to implementation, that is where the problem comes. And again, I agree with Professor Informa that said that you must have the personnel. But in as much as you have the personnel, you should also have good facility to be able to help you implement your protocol in accordance with GCP. Next slide. So, going further, maybe a bit elaborate. We do have what we call an electronic portal. We call it NAFDAC Electronic Clinical Trial Application Platform, ECTAP. That is, it's not complete. I don't know why. That is the electronic portal we use now to collect applications. In fact, it's like an end-to-end process. We receive application from there, review it there to the point of GCP inspection. We have a module for GCP inspection, but it's not yet activated because of some little glitches. But we receive application through the application module, we review it, we give approval, then when you're submitting your progress report, you go through the same portal, submit your report, and we review it. Next slide. So on the front page of the portal, this is what you see. So you can click on this, submit your application. If it's amendment, you go to this module, submit your application for amendment. If it's for import permit, you submit it here. If you are submitting progress report, you submit it on the progress and final reports module. Then serious adverse events, you must report it as well. So you submit it here as well. GCP inspections, we've not activated this module yet. We still do manual. Next slide. So for the application and review, we receive application package through the ECTAP, and which, if you go through our guidelines, you see the components of what you need to submit, which include the protocol, investigation brochure, ICF, and other regulated documents. For Nigeria, we demand you must insure your participants. So you must submit to us the insurance policy as well of the instrument you collect your information. So we must see them too you attach it there and we see it. So the moment the system admin is notified, I'll be notified, then I'll send it to somebody who will now screen your submission and validate all the documents you've attached. Then we send you back payment advice to pay for the processes as well as for the GCP inspections. So if you pay, at the moment you make the payment and submit the receipt, that is the point you activate your 60 days. So you activate your 60 days, then for every process, the system will prompt you. Maybe will tell you that you are at the payment stage, you are at the review stage. Then after that, if there are queries, we will send it to you through the system, then you respond back through the systems. Then, like I said, timeline is 60 days maximum, but like I said, many of the studies we receive, if it is not complex, we go less than that. But there are instances we've gone beyond that because the study is so complex, we need to get expert advice from our expert committee for us to be able to approve such. Next slide. So the review of the protocol is to determine the scientific suitability of that study. Again, we look at the question that you need to address. It must be well defined and well focused. Is it in tandem with the disease on that study? Then we look at specific objectives, like the objectives must be very specific, measurable, achievable, relevant, and time-bound. Again, Prof made mention that you could have many objectives, but you must have a primary objective, the main question. Again, if you can achieve your primary objective, I think you've done almost 50% of it. But it's good if you can go beyond that. Your secondary, even if you have exploratory, go ahead and do it. If you have the means, you continue to do it. Then it must align with the current knowledge. Does the research question align with the current knowledge and understanding in the field? So you must justify that in your protocol. So those are the things we look at. Then we look at the study design. You look at the methodology. Is it appropriate? Do you have appropriate design? Do you have proper sample size that is powered? You must show us how you calculate your sample size based on your disease epidemiology. And you must show us how you collect your data, the collection methods, how you manage it, and it must be well-defined and robust. So it starts right from the time you're screening to the time you do enrollment, and the time you continue the study, pick your data. Next slide. Then we look at the participant selection and recruitment. There must be clear inclusion and exclusion criteria. This will really help you to get accurate data, because if you don't have a clear guidance of the eligibility criteria, you might not pick the right participants to participate in your study. So we need to have it well defined and it's in tandem with what you're studying. Then recruitment strategy, tell us how you're going to get your sample size, because it's not going to be good if you don't have the population that is with that disease here and you decide to bring a study here. Definitely will not get it. In fact, there's what they call clinical trial informativeness. Again, informativeness entails you having robust design from the beginning down to when you get your final data. Again, if everything is clear in the course of design, the sponsor understands what it's doing, the PI is involved in the design and understand the protocol very well, regulators as well, ethics committees, even your participants, you understand what your protocol is all about. Going back to the statistician, your lab scientists as well as farmers, everybody is well articulated with the protocol. Again, that will lead you to a very successful conclusion. And again, you must make sure that you have enough funds that will drive this study to the end. Many of the studies today, somebody was asking the question here that why was it start and stop? Again, many of the studies today, particularly during COVID, many of them were uninformative. I think less than 20% of all the COVID studies concluded successfully. They stopped along the line because they didn't gauge. Some of them, they ran out of funds, ran out of participants, and they couldn't continue. So it's good to have well-informative studies. It will minimize costs. It will give you a very robust data. So you must talk about, you must always very define your intervention. What are you intervening? E.g., are you using drugs for intervention, device, or procedure? Again, even the surgical procedure too, it's qualified for clinical. At the moment, we're overseeing 2 surgical procedures, clinical trials that are ongoing now in the University of Lagos. So those ones too need to be demonstrated. Then you must have an appropriate control group, and you must have good blinding and masking if you are doing a blinded study. Then the outcome measures or your endpoints must be well defined, clear defined, and measurable. You must be able to measure outcomes. Otherwise, you don't make a point. If you do a study, you cannot be able to measure what is your outcome. So you must have appropriate data analysis plan. That's statistical analysis plan. And again, for us, you could submit your study without it. You could give us a brief, but before you close out or before you stop collecting or your data log, you must be able to give us your SAP detailed for us to be able to understand what you plan to analyze and what inferences are you looking at. Next slide. Then your study must be realistic in timelines. Tell us when. If there's need for an extension, you must justify that to extend it. You submit to NAFDAC, we grant you extension. Then you must, like I said, adequate resources to start and complete the study. Not just exposing these participants and you leave them along the way. It does not make sense. So you must have good management and oversight. There must be good quality control or quality assurance during the clinical trials. Because it's of no point if the PI who have the overall responsibility of the study, most of the time it's not there. We went to a study and we found out that the PI lives in Abuja, where the study is taking place in far north. Then what is he doing? He said we should come to Abuja to see him. I said we didn't come there to see him in Abuja. We came there to make sure we see what he was doing at the site. So it's quite unfortunate that the study was also stopped at the point. So there must be ethics committee approval for every study that comes to us. Without ethical clearance, we will not give you approval. I give you an example. We did a joint assessment, African joint assessment of, I think, 8 countries for a study, because when we're doing joint assessment through African Vaccine Regulatory Forum platform, we do it like a country will be together with its ethics committee. So we went together with our NREC, we did the assessment, we came up with all the queries, about 470 queries. We got responses, but coming back home, ethics said they still have had some issues to clarify. And it took them, we're good with the responses, but it took ethics like about 6 months for them to get satisfied with the submission before they approve. So we couldn't have done that because ethics hasn't given us approval. So there must be ethical clearance before we move on to approve a study. Then there must be participant confidentiality and data protection. This must be highlighted in the protocol. Otherwise, it's a red flag. We will not approve it. Next slide. Then we talk about review of preclinical data. Like I said, for every study that comes to us, we want to see the preclinical data. What happened before you started the study in humans? So that will really guide us in what we in what we will see. Maybe in part of in the safety assessment, because there could be something that happened in animal model that if we don't know and it's happening here, we may read it differently. But we want to see what happened in the animal model before it comes to humans. So you must submit to us preclinical data. So it's a very critical step in the development of new product vaccines and medical devices. And the reason for doing this is to evaluate the preclinical data to assess the safety and efficacy of the product under study, to evaluate the quality of the preclinical studies data, including the design, conduct, and reporting. Then to make sure that the relevance of the preclinical data in humans, including potentials for extrapolation in humans, safety and efficacy. That means the safety that you discovered in animals can be tolerated in humans. So that is why we must go through it assess it and give our own opinion on that. Next slide, please. Then we review the quality data of the product under study. Again, for every product under study, it must be able to meet the basic good manufacturing process and guidelines. So we want to see the quality of the product that you want to study. For instance, we data on the chemical and physical characteristics of the investigational product. We will see the data on the manufacturing process, including batch records and standard operating procedure. Again, some people do make that mistake here. If you are conducting a study on an already approved product in Nigeria, they just get to the open market and buy the products. Again, you need to know the batch that you're conducting the study. So we advise applicants, get to the manufacturer, discuss with the manufacturer, let them know that you want to conduct a clinical trial on the approved products, and let them dedicate a batch for you to do this study. Otherwise, something could go wrong. You can't hold any batch as the root cause of your study. I give an example of a study we did, and eventually some people went to court on that. What really safe was the batch that did the study with the product. And when we got to the site, we got that batch, and we brought it, they went to the manufacturer. We discovered that the batch was, from the manufacturing records, there was no issue with the batch. So it's advisable, get a particular batch, do your study with that batch. If there's need for you to add, need for more investigational products, get back to them, let them give another batch, and you have records on that. So we need to have the data on quality assurance, including testing and inspection records of that site, data on stability of the investigational product, because again, you must know at what temperature, what is the shelf life of that particular product. Based on the manufacturer's instructions, you keep the product. That is why we do accountability. And Prof did mention today that we still should get an alarm system. Yes, that is one of the reasons. Because if the manufacturer tell you to keep this product under 25 degrees centigrade, and again, you know, sometimes they have done stability studies. That is why they establish that this is the. So beyond that, for some certain period of time, you begin to lose potency. And you doing a study, you might not get good result on that. So that is why you need to monitor the temperature. So, and it's a good thing if you have a temperature monitoring device that when you are not even there and you have a little excursion beyond a certain limit, you get alert at home and you quickly correct that. The perfect place I've seen it in Walter Reed military. They have a system whereby when there's an excursion of 2 degrees centigrade, no matter what time of the day, they do get alert, and somebody will get there to correct it. So I think it helped them to keep their IP in check. Right. And thank God you did it, right? Yes. Thank you. So you need to have data on the impurities in the investigational product. That will really affect the potency of the product you're under study. So all this information, we must see it and verify them. So upon satisfactory review and after meeting all our regulatory requirements, we can now be comfortable to give you approval. That is, we've established that the safety outweighs the risks. The benefit of the study outweighs the risks. So the approval is given by the Director of Drug Evaluation and Research, and we keep saying upon approval, the sponsor or the applicant need to inform the agency 2 weeks before commencement of the study. They will really help us to control what is happening. So, and be noted that approval is renewable every year. Next slide. So, after you get the approval, the next stage is for you to get the license to bring in your product if your product is going to be imported. But if your product is manufactured in Nigeria, like I advise, get to the manufacturer, have a discussion with him, have a dedicated batch that you do this study. But for those who want to import their product IP into Nigeria, they need to apply for an import permit. And again, you do this through the portal. Again, the process involves putting your application through the system, the portal. it will be screened and validated. All the submission, we have a guidelines, I told you. You go through the guidelines, then come here, put in your application. After we screen and validate your documents and it's good, you pay fee for the processing. Then we assess your submission and we now approve your submission. Again, some of the key documents will involve the product dossier that the IP dossier that you're going to do the study upon. Then if it's already a registered product, you need to have a dossier. But if it's a new product, you need to have brochure. That's all the subsequent researches, publication are there. The quality of the products at that point of development is in the brochure. So you submit it as well. Then you need to submit an approval letter because you must get approval before you apply for import permit. Then you must show evidence that the site producing that particular IP has gone through current good manufacturing practice compliant. So you submit that to us wherever the product is coming from. Then the labels must reflect what is in the guidelines in accordance with the IP labeling. So we do advise applicants to submit, as you're submitting the CTA, submit the application for import permit. Again, to minimize turnaround time, so that at least the moment we are approving your study, we know we've already submitted the application for IP, we review it together and give you approval. And the maximum time is 15 work days. Next slide. So, and when we approve the study, we need to monitor what is happening because they say you have the right to hire and fire as well. So we monitor what is happening, and there are many ways to monitor what is happening during clinical trials. So some of the ways we do is to review study progress report, which will comprise the overview, progress overview that reflect the snapshot of the trial status, including enrollment numbers, completion rates, and other information regarding the study. We will know what is happening, and this is seen when you submit quarterly progress report. There we will see how many number of safety adverse events you've picked, serious adverse events you've picked, how many have withdrawn from, you've lost to follow-up, how many have left the study, how many you've screened, enrolled, all those information will be there in the progress report. And that will give us confidence that it's been done in accordance with the regulatory requirements. Then we'll look how you adhere to protocol. Then we'll look at how you report your safety information. If there are safety signals, how many have you recorded, how many have you reported? We're not expecting you to submit adverse events report to us. When we come for inspections, we see it, but later you can submit the line listing, as some of you do submit. Then, but the serious adverse events must be submitted to us. Otherwise, it's a contravention. Then we look at the data quality, the quality and integrity of that study report and evaluate it. Then we know whether you are in line or not. Next slide. Then we do what we call on-site inspection. As part of the monitoring, we do GCP inspection, inspection at the site to ensure compliance with regulatory requirement. And we monitor your SAEs, because if we submit your SAE and with your causality assessment report, we review it. If there's need for any doubt, we will promptly go to the site to see what happened and to verify it as well, what your report is all about. If we doubt your report, we come in there and see what happens. Then we review your clinical data time to time. Again, when you have any data safety monitor that have come to review your study, again, when we come there, we see the reports and your monitor report as well. Next slide. Then, at the end of it, we expect you to submit to us your end of study report after completion of the study. And it's a comprehensive document that summarizes the results, conclusion, and implications of the clinical trial. Again, we require all applicants to submit end of study report for all approved clinical trials through the CETA portal using ICH E3 guidelines. This is a guidelines that we've adopted that provides guidance for the structure and content of a report format. So we employ our stakeholders use that guidelines to design your report. And the report is submitted within a year after completion of the study. So the importance of this report is to make sure it creates transparency, accountability, and this will contribute to medical knowledge because, of course, there's a reason why the study was conducted. So the end of it will contribute, we believe, to the medical knowledge, whether it's successful or not. Then will help to inform decision by healthcare professionals as well as patients and regulatory authority. Then it's part of regulatory compliance. You must submit to us. It's part of regulatory compliance. Next slide. Then the key component of the study report, end of study report, you submit information about this trial design and methods. You describe it and the method you use to generate those data. You show us in a pictorial format the participant flow and baseline characteristics of the participants you recruited. Then you show us your efficacy and safety results that you generated from the study. Then you show us all the adverse events and the serious adverse events captured during the clinical trials. Then you have a discussion and conclusion section that you let us know. Then your recommendation for future research. Then all your appendices that contain some of those tables and other results that will really boost your report. Next slide. So, and after the review of the study report, we must give our verdict. So, the report must be complete and accurate, must be clear and concise, consistent with regulatory requirements, and there must be trial results and conclusion. Next slide. So when we review the report, it could either be accepted or rejected. So if the report is robust enough and answer the questions that it was designed for, is good for us as well as for the researcher and maybe for the participants. But if it did not meet those basic requirements, it can be rejected. Then the consequences of non-compliance with regulatory requirements are rejection. We can reject your study report, and we can delay or reject your application for marketing authorization because we may need certain clarification from your report. And sometimes it could lead to regulatory actions, which could be an enforcement of the regulations that we have. Next slide. Then finally, again, after we've approved the study, we must get to decide to see what happens. And the essence of GCP inspection report is to make sure that the study is being conducted in accordance with the approved protocol, in approved in GCP guidelines, ethical guidelines, as well as making sure that you meet the basic, you protect the well-being and of the participants. So it's defined here as the act by which regulatory authority conduct official review of documents, facilities, record, quality assurance, arrangement, and any other resource deemed necessary to the clinical trial, and they may be located at the site of the trial, at the sponsor site, or contract research organization facilities, or at other establishment. Again, at the moment, we have not gone to any sponsor site to inspect, no CRO site, but mostly our inspection is based on the principal investigator's site. And we've come here several times. Prof is here. Next slide. So, the goals of inspection is to make sure we safeguard the rights, safety, and well-being of those who are giving you the information in the trial. These are guys that are giving you free information for you to achieve your desired objectives. So, we must make sure their rights, safety, and well-being are protected because the drug can harm them. So we make sure it's not. Then, want to verify the quality and integrity of the data produced from that study. We want to assess compliance to the approved protocol and regulatory requirements, to assess availability of quality system in place during the conduct of the study, then to identify areas of quality improvement. Again, I keep saying we go there not as policemen, but to make sure we improve the quality of the study so that data coming out from there, we'll be proud as well if we get robust data coming from Nigeria. Next slide. So, it's only scope of inspection inspection applies only to clinical trials authorized by the agency. Those who are not authorized, the moment we find them, we will sanction them. We have the rules and the facility to sanction them. Either take them to court or we bar them from clinical trials. So site can be inspected before, during, and after the conduct of clinical trials. We can do pre-conduct, during the conduct, and post-conduct. All phases of clinical trial can be inspected, including bioequivalence studies. Next slide. And we do this on a routine basis. That means we schedule the inspection and we inform the site that we are coming to inspect. Then we get the consent of the site, the PI, his readiness and his team before we get there. Then it can be followed up. If we've gone there before and we give you compliance directive or CAPA, then if you respond, there's need for us to come, we come for follow-up to see whether we implemented it well or not. Then we do focus or triggered inspections. When there's need for us to come, maybe we'll get information from somebody that something is happening somewhere without regulatory approval. We rush there to see. Or if we receive safety, serious adverse events, we review it and there's need for us to doubt maybe the outcome of the assessment. We get to decide and see what really was the cause. So that's what they call focus. There's a reason for us to go there. So they can be announced or unannounced. That is, for the focus, we may decide to announce it or not announce. Next slide. So, and we do this inspection on risk-based approach. Again, the reason because at the moment we have so many sites, I think we have close to 100 sites at the moment. And if we look at the manpower as well, we have, it's not enough. We look at the financial implication as well, might not be able to cover all sites. So for instance, if you have 12 sites, we can rank your sites on the risk-based approach and then pick the sites and we can come in to inspect. And the criteria for those selection is highlighted in the next page. But then we classify them into low risk site, medium risk, high risk. And all these are based on criteria that could either affect the risks to participant safety, data credibility, or smooth implementation of the study protocol. So the criteria could be the phase of the study. Again, we prioritize phase 1 study, which is very critical, than phase 3 or than phase 2. So if we have phase 1 and 2, we'll choose to go to phase 1 first before phase 2. And the characteristics of the IP. For instance, if we have biologics, which is very sensitive, or we have vaccines, we may choose to go there. Or you have monoclonal antibodies against the normal small molecule, we may decide to pick all those heavy molecules to go and see what is happening there before the other one. Next slide. Then it could be based on suspected fraud or misconduct. If we hear it, we quickly get to that side. We choose that side first. Then if there's a failure to submit quarterly study progress report, we know that something might be cooking there that might not have given you confidence to submit your report. We come there to see what is happening before any other site. Then if we receive frequent essays, again, we know maybe there's a breach. Either could be from the eligibility criteria, or maybe the personnel are not well trained to implement the protocol in accordance with the principles. Then, if we have various variations in applications, again, there was a study we had that we won until the thing got killed. So you keep changing PIs, you keep changing sites, you keep changing goalposts. We come there to see why is that happening. Then, if you have deviation or violations, particularly violations, we come there to see why it's recurrent. Then, amendments as well. Then, we do follow-up on non-compliance. For instance, we've come to your site, we give you compliance directive, and you refuse to respond. We have the need to choose your site first before any other site. Next slide. So, like I say, we investigate the PI site. We do manufacturing site for investigational product, and we inspect the lab. That, for instance, if the lab is not domiciled in where the study is taking place, no matter where the lab is, we have the prerogative to go to that lab and inspect it. Because, again, the outcome of the lab form greater part of the outcome of the study. So we can do sponsor or CRO site. Next slide. Then as we prepare for inspection, We plan it depending on the scope of the inspection. We notify the site PI or the sponsor. Then we get confirmation from the sponsor or site. Then we prepare and go for the inspection. Next slide. Then at the inspection, we do what they call opening meeting. We discuss with the PI with his team. We get to know a lot what is happening. They ask the question they want to ask. And we do the real conduct where we visit all these facilities. Then at the end of the conduct, we do what they call close-out meeting, whereby we enumerate what we discovered. We give the PI opportunity to respond to defend some of the things or provide more evidences. Then at the end of it, what we agreed upon, what they cannot respond or what they cannot be able to justify, we carry them as our findings. And that is what we're going to report back to the site PI. Next slide. So, like I said, I've already talked about it in the opening meeting. Next slide. Conduct. Next slide. Then closing meeting. Next slide. Then these are the sites we inspect. We inspect the organization and personnel of the study. Facilities and equipment, organization and documentation, very key. Monitoring and auditing, very key as well. Use of computerized system, informed consent process, source document verification, efficacy and safety assessment data. Then we look at the IMP handling. That is where the issue of accountability comes in question. If you cannot account for a tablet of one of the IP, it's an infringement, which definitely you must justify that. Next slide. So, and after the inspection, we'll classify our findings based on some certain criteria into critical finding, major finding, and minor finding. And for critical finding, these are conditions, practices, or processes that adversely certainly affects the right, safety, or well-being of the subjects and or quality of or integrity of the data. This includes pattern of deviation classified as major, bad quality of the data, and or absence of source document. That's an example. If you don't have source document, where did you generate your document in your data collection forms? So source document, you don't have it, it's bad. Then if you have major findings or non-conformance, these are conditions that might or process that might adversely affect the right, safety, and well-being of the subject or quality of the data, right? So you must avoid having so many majors, because if you collapse the majors, it can lead to critical. So most of the times, majors are the ones that drive a site to critical. You might not, ideally, if you get a critical finding, even one, that study is supposed to be posted, depending on the situation. You can post the study, you can recall the products, you might stop that particular maybe participants who the critical condition happened from taking the IPs for some time. And minor conditions are practices or processes that will be expected to adversely affect the rights or well-being of the participants as well as the quality of the data. So we always want to see minor infringement, but again, if we begin to discuss our experience on the field, we are yet to get there anyway. But I believe with this type of events, gradually we will get there. Next slide. So expectation after an inspection. We classify the observation in the written report. We send a copy of notice of findings to the PI at the site. Then we all the observations, and we give the investigator a CAPA plan, that's corrective action and preventive action template for you to respond to those observations. Then when you respond to us, you send to us within 30 days. We review your CAPA submission, and if it's good enough, we may decide to close out the inspection with a written letter. But if there's more need for information, we get back to you again and till you finally close those CAPAs. Next slide. So, finally, like I said, we have the hammer with us. We can penalize, So, and part of the sanctions include given timelines to address issues. We can give a warning letter depending on the severity of the infringement. We can stop the study or recall the product that is already in on the study, and we can suspend the study. We can even reject your data. In fact, at the point we can prosecute a site, a PI, or the sponsor. So I think that is the end of my presentation. Sorry for taking your time.
Please, let's appreciate Dr. Beno Yakubu once again. Thank you so much. We had to give him that time because he's a regulator, and it's important to hear from the regulators. Okay, so we'll hold on with the questions. We'll take the next presentation, and that next presentation is also from the regulatory aspect. We want to hear from the chairman of the Health Research Ethics Committee of our institution here, in the person of Professor Issa Yabaji. And I want to also let everyone know that he's wearing two caps here because he's also the Deputy Provost of the College of Health Sciences, University of Abuja, and he's here representing the Provost. You're welcome, sir.
Thank you, participants and the organizers. I think my job has been made very easy by the just immediate past speaker. He has summarized my presentation, and therefore I will not take more than 10-15 minutes. Yes, University of Abuja Teaching Hospital Health Research Ethics Committee, otherwise known as UATH-HREC, is the organization that I chair, coordinate with very eminent personalities. And we went to, in this presentation, shows you how you will scale through easily. Ours may not be as rigorous as NAFDAC, but we are very close to that too. So let's go into the case. Before this, we will center our presentation on how do you get away with the procedure of applying to our RRB. The consent form will be mentioned, and then we will speak a little about the material transfer agreement as it applies to some researches. And therefore, take-home messages will include, but not limited to, the following. How to get certified for human research is the basic standard that all researchers must have before they venture into any human research. And we expect you, we shall guide you to a link where you should have that. It's free of charge. You get that, you don't pay dime, and you get well educated. And then we shall acquaint you at the end of the session, step by step, how you scale through our ARB procedure, and then the importance of MTA, as we have said. Yes. The NAFDAC presenter has said it well, that our aim of the regulation in our ARB is to ensure that our subjects are not taken for granted. Their rights are not violated. When it is necessary, the researcher will have to take responsibility of whatever happens to the patient along the way, and we do receive frequently cases of mishaps, unwanted effects, And that also leads us to it, depending on the degree, as it is said now, whether critical, minor, or major, for us to stay, hold on with your research. And then we'll look at it again. And we keep the record of all past researches and the ongoing researches in our facility for higher authorities to review. Straight to the point, we are grade B, IRB, and by that it means if you have a research that will cover no more than 2 states, we will be able to give you the research, the approval. However, if your research is spread across Nigeria from 3 and above states, you need to possess an NREC approval. The link to apply to our IRB is posted, www.rips.com.ng, and you are there. The procedures are self-directed. Just fill the form. You get the link will be sent to your email, verify it's you, and then you carry on with the process. What do we need when you are submitting to us? You must give us a highly summarized CV, no more than a page, of who you are, where do you work, what have you done in the research field, so that we'll be able to determine whether you truly be able to carry out what you're about to say. Whatever you have submitted on online must be on up. We have seen cases where a proposal is sent to us, particularly the academic ones. The owner does not append his signature. The supervisors do not answer up to the research. And that begins the beginning of our problem with the candidate, because we don't go forward from there. If there are letters of support to co-investigator by the principal investigator who may not necessarily be residents in Nigeria, that needs to be presented. We have seen a case. What we are referring to by this point is that if you have an international collaboration, you are not the principal investigator, but the research is to take place in our hospital or within Abuja and any other state around us, and we do not find a co-PI in those stations. Of course, who do we hold on when there are problems? That we find it very difficult to approve that research. Sponsors attestation statement, where applicable, must be uploaded. And then a special application, 2-word, 3-word, that you are the one applying for this ethic approval, and then we take it off from there. That must also be signed. What you are uploading must have a captivating title, a title that speaks for itself. We are beginning to see from our young researchers titles that are paragraphs. And therefore, from that stage, you're already setting in a confusion. So your title should be smart and should be able to lead us to where we are going. While you are attempting to introduce what you're about to take on, you should also show us in your literature review. You should also show us in your literature review what you have done in the same field before now. Whether you have partaken in any previous research that has ended well or not. Show us your previous work. Then the purpose of research should be shown. The aims and objectives, as said by our last speaker, must be smart and achievable. If the aims stated there by your standard, as we have seen in your resume, you will not be able to achieve that research, then we'll not go ahead to give you the approval. You must have the ability, and therefore your aims must be the ones you should be able to achieve. You are not necessarily trying to get an international recognition from the research you are doing. So know your level and cap your research objectives to those that you can handle. The materials and method, and I've started that place because that is where the problem is. What methods are you going to use to collect your data? How did you get your sample size? Are you using a prevalence sample size formula for a comparative study? And you must reference them. What is your sampling method? Is there likely going to be an injury to us participant subject? Those are the areas we spend our time to review and review very well to protect the subject that we participate in the research. The costs of the source of funding for this particular research must be explicitly stated. What is the source of your funding? What is the size of the funding? How are they allocated to various things? Is there a space for due compensation? If there is, we must know. If your participants will have to be traveling to a research site repeatedly, who pays for the research? Who pays for the travel expenses? That must explicitly be stated. We also want to know, from your funding, suppose there are unwanted side effects, will you leave the patient or the subject at his or her own peril? Or will you take care and correct the anomalies that are setting for the patient? This will have to be seen. In your cost of, the details of your cost, we should also see evidences that you are managing the resources effectively as stated out by your funders. Those are some of the things we also looked at. And if you fulfill those righteousness and you do them the way they are supposed to be, then you'll be making our job easy, and you should be able to get your approval in the shortest possible time. Consent form is another area after methodology that we waste our time, we spend our time looking at. Consent form has been sent to us that simply says, I am Mrs. ABC trying to carry out this research. It will not be harmful to you. No signature, nothing. That is not the aim of the consent form. The consent form must be self-explanatory. The terminologies you are using for some of our participants that are likely to be laymen must be at the level of understanding of a GSS 3. By the time you begin to talk about the pathogenesis here and there from your consent form, there's no how that your participant that is not properly lettered as you are will go along with you. The participant must know what you are about to study, what is the advantage, how is he going to benefit from it, is he likely to have benefit for him or her or the later generations. Those must be explicitly stated. The clarity of the presentation is needed, the depth, the details of how you are going to collect the samples in summary forms has to be there. Are there going to be compensations when it is required? And how are you going to take care of the risk must also be stated in your consent form. And you must, at the end of it, endorse. There should be a place for your signature and the signature or a thumbprint of your participant. When these are not there, then that may be the source of rejecting your proposal. The summary of ethics, when we look at the ethic issues within your proposal, this must be followed. This is known to every researcher. What you must not harm the patient, the participant. If you cannot do any good to him, leave the patient as he is. Your drugs or whatever you are using for the research must not be harmful to the patient. The purpose, the respect of his or her dignity must be repeatedly adhered to. The non-maleficent, there's no research that does not have harm. Because even if the candidate is only filling your questionnaire for you, he or she is spending some hours there or minutes. And those minutes can never be gotten back. And therefore, there is always a risk that is attached to a research. But what we are after is that the harms contained in your studies are no more than minimal risk. And that will be acceptable. So writing in your consent form to say that there's no risk does not hold water. Even if you are doing a retrospective study, what are you going to do with the data you have collected? Are you not going to expose the owners of those data? How are you going to protect those data? And from there, we may get either, no matter how little the risk that is associated, you must have fair recruitment of all participants. You give them equal chance of being chosen at an appropriate based on your guideline. And when your research is garbage in, garbage out, of course, that will attract comment from us. We've said the result of earlier related studies, like your phase 1, phase 2 studies, if they are available, that should be indicated. We should find them within your research so that we'll build up on that and knows that you are going to somewhere. Questionnaire, performance, and other relevant research tools must also be attached, and we do screen this also. Because if you are studying the effect of ACT, for example, on a particular population, whether the person is black or white, provided they live in that community, may not have a significant bearing on your research. There are questions you ask in your questionnaire that you are wasting our participants' time. If the questions are not necessary, do not ask them. So we look at them very well. Complete, and then particularly, you must be anonymous in your questionnaire. You can identify them by some keywords that is only known to the researcher, and then you are assured of the safety of those research, rather than you are a pro forma containing places to be inserted the names. Complete material transfer agreement. Yes, if the study has collaborators outside this country, and it will involve the movement of the document, the informations, transfer of samples, storage of samples, and the analysis of these samples outside this country, then there must be a detailed material transfer agreement. If the protocols are available on net, if you do not have them, and during our monitoring we realize that you are transferring your material to outside country without an MTA, then you are liable. We stop the research and then begin to ask you questions and probably may even refer you to the bigger controller, the NAFDAC, that this person, even though we have given him the approval, he has not disclosed maximally what he ought to have disclosed to us. There are cases that you need to give us a plain language summary of your proposal. Plain language summary, or at least of your consent form. Where you are using English and the candidate cannot understand English, then give a summary in the language the patient can understand. You may use an interpreter, and that will be acceptable. Informed consent, we have said that it should be at the level that a JSS3 should be able to understand. Then you'll be passing information. But if you are using grammatical terminologies that is difficult to be understood by a JSS3, then you are not sending any information. Any other document that is considered relevant for the ethic review certification, that should be submitted. Our procedure in UATH, we sit statutory every last Tuesday of a month, between 1:30 to at most 3:30 or 4, depending on when we finish. We require you to submit at least 2 weeks before the scheduled date. So if we are sitting, the last Tuesday of the month is 29th, and you are submitting on the 18th, your submission will not likely be taken that month. We are not necessarily, by this, we are saying that you have the tendency to have your results if you submit at least 2 weeks to the scheduled date. But there are cases like most of the researches that our professor here submit, that one can drag us to 2 months, 3 months, even though we stretch ourselves occasionally to produce the result within a month. Because majority are heavy collaborative studies, having international component, having to do with phase 2, phase 3 studies, and therefore that we cannot run over within a single sitting. There are cases that we have to invite the researcher to come and explain to us in the flow of the meeting, because the challenges are so enormous that writing back and front will take our time. The admin usually distributes whatever has been uploaded into our site, to various reviewers. And then we give them a maximum of 2 weeks to turn in their report, at least before the day of the meeting, or come along with the meeting with the report. Then the full committee will decide on each submission that constitute 14 to 16 members, led by the chairman and the specialists from various departments. Because if the chairman is a pediatrician or gynecologist, if you submit a research that has to do with hematology, I need someone that knows the ABC of that area. And this also includes religious representative, the community leader representative, market women representative are within our sitting. If they are not going to be there, they will take an excuse. And if they come and are not later, we must translate to them whatever we are discussing, so that we carry them along. If you are taking sample is, for example, or you are spreading questionnaire within the market, and there's a market woman leader there, you may be approached and say, well, which letter are you distributing and getting information from our people? But if our representation is within our ethics committee, then that saves you that embarrassment. There are cases that we may exempt you from a review when we realize you are dealing with no more than minimal risk in a retrospective review, and the safety of the data is assured, or we may occasionally grant you accelerated review depending on the extent of the risk contained in what you have submitted. Outcome. At the end of our sitting, the admin will inform you. If we find your submissions to be in line with our standard, we'll simply give you the approval. However, for some, there may be further review. Candidate is contacted by email, and then 2 options. We will simply say, grant the approval if he has sent back to the admin and has answered all our questions appropriately, and then endorsed by the chairman, give the approval. May not necessarily wait for the next sitting. Or the candidate will be required to resubmit completely for a fresh review if he has derailed completely from the set line objective. When there are major deviations, of course, he has to resubmit. There are cases where we completely decline or reject the review because the candidate does not have the capacity of what you want to do. We have received a proposal from this university, from someone that does not work within the hospital system, who wants to be taking the blood samples and to be reading it, and we do not find any microbiologists in the as part of the core investigators. The site of where these readings will be stated are not there. Well, your cost does not go close to using the blood sample. So in that case, we simply reject it, and you come over to us and we tell you to go and write it appropriately, if it has to be done. The rules for renewal, if we have granted you approval, our ARB and virtually almost all requires that you should there is a room for renewal of your ethic approval, and your submission must be within the last 4 weeks to the expiration of the current license you are holding. If you submit out later than with less than 4 weeks to the expiration of your something, then we will not renew it for you. You may have to re-upload and apply fresh and pay the appropriate charges. We take this for serious. So we have gone around and briefed you about how to submit, what to submit, how to expect a quick response, and what not to do. If you do not have the capacity, if you do not have the whereabouts, do not submit to us. So we said your topic must be self-driven. In concluding, detailed budget consideration is vital. Performing topics are often easier to defend, and ensure you and your co-researchers own up the project by endorsing it appropriately. Meticulous submission ensure fast and accelerated approval. I thank you for your time and interest in this. Thank you.
Thank you very much. Please let's appreciate Professor Isaiah Baji once again. I know there were a couple of questions online, especially after Dr. Beno Yakubu's presentation. We will handle that, so please, the online team, just hold on for us. I want to introduce the next person. He's somebody who needs to talk to us about bioethics. He's going to talk to us about clinical trial ethics and human subject protection. I must say that, please permit me, and I know why I want to do this. I want to appreciate Professor Adebayo Adejumo for coming at this point to do this presentation. So, Prof, if you're online, they will unmute you, and then your slides should come. Okay, the slides are up now. So he's a professor of health psychology and research, and he's a faculty member of the Center of Bioethics. Prof had mentioned briefly about that Professor Noda, about Professor Adebamowano. He's part of that faculty, and many of us, the certificates we hold in terms of good clinical practice and all that comes from that center. So please join me and welcome Professor Adejumo.
One in the Bible, you can find that there was one, investigator. Although the Bible didn't tell us that this is a clinical trial that is running in Daniel chapter 1, it only talked about, and that Nebuchadnezzar wanted to enroll Hebrew children. He wanted to make them capable to stand before the king, to understand his dreams, and to be able to proffer solutions or answers to the dreams that we will have. But one thing he didn't know was that he said that the clinical trial that he wanted to do, he wanted to give them king's rich diet. That's what we were told in that clinical trial in the Bible. That he was going to give them king's rich diet. And the study was going to last for about 10 months or so. But Daniel, who was a member, also fitted into the eligibility criteria in that clinical trial in chapter 1 of Daniel, raised his hands, troubleshoot, did a little bit of whistleblowing, and called the principal investigator and the co-PIs. Oh, look, man, even though you are bringing an intervention to achieve this purpose, he said by virtue of his own academic dexterity that he knew what could be done better than the proposal that was floated for that clinical trial, for that research. He suggested that he would just take legumes and water, not only for 10 months, but just not even for 3 months, but for just 10 days, and that the research purpose and objectives will be achieved. And it will be surprising. The principal investigator now got to know that this man must be speaking from a side of perspective of information. So you claim that as a principal suggested by a potential research participant, that's Daniel in Daniel chapter 1, produced a better result and with a shorter duration. And of course, in ethics, there's something we call principle of prudence. In principle of prudence, you should use the minimal resources to get the overall realization of your research objective. There's no point if you tested your research hypothesis where you are projected that will be recruiting 35 people, but at the end of 27th recruitment, and you have been testing all along, and your research questions and hypotheses have been sufficiently and consistently confirmed. There's no point recruiting the remaining 6-7 people because you'll just be exposing them to undue risks. So Daniel brought that up. So we also know in our time that when we are floating clinical trials, research activities, that some of those people that we think may not know so much may know more than us. So still within the ambit of informed consent, we need to share what are the procedures, what are the benefits. And this is very important because it helps us to also ensure that the autonomy and the autonomous right of those who are potential participants are protected. And when we do this, this will protect responsible conduct of research. And in my slide here, I called it transparency, because transparency is an ambit. It's a subsegment of responsible conduct of research. And of course, it also helps us to put our scientific validity to test. Any study that is scientifically weak can never be ethically sound. So even at ethics committee, when we pick up protocols and we're reviewing clinical trials, one of the things we look at is the scientific validity of that proposal, the extent to which, the manner by which you want to answer your research questions will be realistic based on the method that you have suggested. So clinical trials must be done methodically and scientifically sound to justify exposing participants to any form of risk. And you need to know that poorly designed studies are unethical as they waste resources and may harm participants unduly. And of course, by so doing, they're not going to yield any benefit to the research community. Okay, let's go to the next slide for the reason of time. So the next thing we need to consider ethically in terms of the principles that should guide what we do as clinical trial specialists is that we must be careful to select subjects fairly. You don't subject people into the harm that you know will be safer, will be more beneficial by virtue of somebody's being related to you, or maybe a member of your old girls association, or old schoolboys association, or a member of your church, or a member of your mosque. That should not be the criteria for determining into which of the arms of the clinical trial your subject should be selected into. There must be fairness in the selection. In other words, we talk about characteristics of scientific experiments. For anything to be scientific, for anything to be ethically sound, it must have, or it to be an experiment, there must be introduction of a treatment, that means an intervention, there must be randomization, and there must be control of extraneous variables. With that at the back of our minds, when the study proper is about to begin, the manner by which you put people into each of those trial arms also should be based on justice, fairness, okay? And of course, it should be based on scientific objectives, not according to people's convenience or your own convenience or the vulnerability that you are perceived. So this prevents exploitation and promotes equitable access to research benefits. And the principle of a favorable risk-benefit ratio requires that potential benefits outweigh foreseeable risks. And of course, it's not the first time that we'll be hearing this, even as early as 1990s 1947, the Nuremberg Code already spelled out, apart from saying that there should be fair subject selection, it also talked about benefits should outweigh risks. So we also must factor that into what we also marshal. And of course, that was advanced in 1964, while the Nuremberg Code was promulgated by the World Medical Association. There we got to know that it's not just about informed consent. We must now do a titration, a comparison of the risk and the benefits. And we're told that the benefits should, the pendulum should swing towards the direction of the benefits. In other words, it should be that the benefit outweigh the risk. But may I say, as people like David Wendler, as people, a couple of other folks at University of Toronto Joint Center for Bioethics will also argue, because these are the specialists when it comes to risk benefit algorithms. They will say that one single risk can cancel out as many as 100 benefits. A clinical trial that has a 33% risk that will attend it is ab initio say that for every 100 people recruited, at least a third of them will die. Built enough mechanisms into the protocol to ensure that the probability of the risk in your clinical trial is reduced to as near zero as possible. And we also must ensure that the benefit that you have, flagged, that you have said you are going to assure, that the extent to which they will be realistic will also be determined by a good, experienced research ethics committee members reviewing that protocol. Next slide for time. So all these things are to ensure that participants are safe and that the safety of those who participate in trials are prioritized. So the next thing is independent review. Independent review was marshaled in 1964 by a Helsinki Declaration. Of course, I know there are very many other documents, like the SIOMS, also especially the 2016 version of SIOMS. SIOMS is Council of International Organizations of Medical Sciences. And of course, if you go to, it's about 120-page document. It's one of the documents, regulations, guidelines I love the most. Not that I don't like Nuremberg Code, not that I don't like a syndicate declaration, not that I don't like the National Research Act, not that I'm not, a supporter of National Code of Research Ethics. I must also let you know, as early as 2004, 2005, I've been drafted into the group that eventually developed the National Code for Research Ethics for Nigeria, until eventually it got published in 2007. Okay, so independent review by ethics committees say specifically that there are people we call IRBs, and I need to say this not necessarily to castigate others, but in Nigeria we do not use the terminology IRB. IRB is American-centric, and of course in other parts of the world, in Ghana, they have VRC. In Canada, you have REC, Research Ethics Committee. They have different names that they call IRBs elsewhere. But in Nigeria, what look like what you call IRBs are HRECs, Earth Research Ethics Committees. That's the name in Nigeria, okay? I specifically put this in this slide so that they can also, drum that loud at this point. So, Research Ethics Committees provide oversight, ensuring that trials meet ethical standards before proceeding. Our friend from the National Hospital already said that, so I will not reinvent the wheel. So also we need to have respect for those who are enrolled to participate in trials. And one of the ways by which we protect those who participate in clinical trials is to be familiar with the principle of privacy and confidentiality. People oftentimes mix those two things together. Privacy is not perfectly the same thing as confidentiality. Privacy is the extent to which I'm willing to share my personal information with someone else, releasing that part of my data for someone else to have access to that information. That's privacy. But ensuring that no one have access unduly to my information, every bit of what I do with that bit is within the domain of confidentiality. And there are several techniques by which you do that. You can save your information in the cloud that no one can have access to it. You can save your clinical trials data on clinical trial registries, repositories that are locked up in filed, locked up cabinets. It could also be saved in passworded computers. And of course, it could be encrypted. There are several methods. You could do anonymization. You could do de-anonymization. These are techniques of ensuring or maintaining confidentiality. And of course, apart from ensuring that, there should still be a caveat that people should be able to withdraw from any clinical trial at any point in time. But you also need to ensure to say to those who withdraw that whatever information that they have yielded along the line will not be retractable after yielding that information, to the research team. In fact, in some instances, some studies have publications that are rolled out before the end of a study. So if such data are form part of the data upon which such publications are made, so how can they claim that they want to retrieve their information? So, but they can say no henceforth from the point at which they feel, for one reason or the other, that they are no longer willing to continue with the clinical trial. And of course, ethics committee play a vital role in ensuring that all these things are written in black and white in research protocols. And not only that, ethics committees have a duty of what we call oversight. It's not just approving a clinical trial. And we need to ask ourselves, as researchers, more essentially as ethics committee members, to what extent do we tiptoe to research sites to ensure that every bit of what we have approved in a research protocol is what is actually actively being executed at a research site? And of course, ethics committees are charged with the responsibility of evaluating research protocols and to ensure that these things are done meticulously, to ensure that methodologies and risk mitigation strategies are not only written down, but we have an assurance that it will be implementable based on the experience, competence, CV, resources available to the research team. So their deliberations hinge on ethical principles such as beneficence, non-maleficence, autonomy, justice, ensuring that research endeavors adhere to the high standards of ethical conduct. And it's from this point in time now, I'm shifting position from the paper presented by Sickle Emmanuel in What Makes Clinical Research Ethical, as published in the New England Journal of Medicine, 2000, and the Journal of American Medical Association, I think in 2004, that's the publication. So I'm now switching over to a principlistic approach, the role of principles. And when we talk about role of principles, essentially, we are talking about beneficence, non-maleficence, autonomy, and justice. I need to say that there are several other, moral philosophical approaches by which you can, talk about clinical trial ethics, research assurance. But because that can take us 3, 4, 5, 6 lectures. If you are talking about the deontological approach, it's saying that as researchers, we have the utmost responsibility, that's our duty to do the right thing, and that our intentions, that's going to Kantianism now, propounded by Immanuel Kant, saying that as much as possible, we should ensure that our intentions matter more than even what we see as consequences. But you'd see people like Kenneth Arrow and a couple of other people that are utilitarian, they'll quickly come to query the Kantians, say no. It's not in all instances that you say that your intentions are better, the denominators of the moral standard or moral value of your clinical trial. They're saying that your intentions don't matter as much as the consequences of your action. In other words, that even if you claim that you intended to do what was good, if the outcome is such that the end does not justify the means, then you are not ethical. That's to a Kantian. And of course, that's to a consequentialist. But of course, there are other positions that are neo-consequentialists. And for example, utilitarianism. In utilitarianism, we talk about the need to consider the greatest good for the greatest number of people. I don't want to go into moral philosophy today because that may not be the full reason for which I've been invited here to speak. So I'll rather focus on the principlistic approach, which came about 60, 70 years ago. And it's an effort to correct the imbalances in other moral philosophical positions that have been applied in the domain of science. And people did say that it's nice for us to know that for anything to be ethical, the autonomy of those who participate must be assured. the benefit must be incontrovertible. Then no harm must be done. That's in the normal efficiency. And justice, talking about the fairness. In other words, treat equals as equals and unequals as unequals. So that's what I want to say very, very quickly now. I'm raising against that. Next slide, please. Am I still audible? I want to be sure that I'm audible. Am I still audible?
Yes, sir. Yes, sir.
Can you still hear me?
Yes, sir. We can hear you.
Okay, thank you. Okay, thank you. Okay, so I want to briefly talk about autonomy now. Autonomy is a combination of two words, auto and nomos, self-rule, self-governance, okay? Somebody is saying, I never viewed the scripture in Daniel from that perspective. I will attend to you, Nora Akinola, later on. Okay, so participants must be fully aware of the trial's purpose, procedures, risks, and benefits, and must voluntarily agree to participate. In other words, you give people the right to say yes or no based on their preferences, based on their values. Okay, whatever they hold as being something cherishable, let them base their decisions based on that according to their preferences. So don't force anything down their throats, and don't coerce people into participating in clinical trials. The principle of autonomy says the recruitment, the informed consent process is that which will guarantee or assure that those who say yes or no, they are saying it based on their personal choices. And of course, this safeguards individual decision making and respect. In other words, it's a technique, it's an effort to respect the autonomous right of those who participate in clinical trials. Next slide, please. Next slide, please. So when we say beneficence, beneficence means even the clinical trial that we want to conduct should add value. So if there's a study that is to be done and it's not going to be of benefit to those who will participate, that study is ab initio, dead on arrival. In other words, beneficence requires that researchers, clinical trial specialists should maximize potential benefits for participants and society. But may I say, it's not in all instances. Those who participate in studies will be the ones who will benefit directly from a study. Okay? There's a saying in my part of the country I come from, that's in Yoruba land, those on whose heads coconuts are broken may not wait to eat part of the coconut. Any interpreter in the house, you can help me to do the work of interpreters later. In other words, some of those who participate in clinical trials, they may contribute to knowledge and lead to the generation of development of new treatments and approaches, but really, they may not be direct beneficiaries. Some can die in between clinical trials. Look at phase 1 clinical trials, for example. Not as many as were enrolled in phase 1 clinical trials may survive it. So in such an instance, we need to let people know. So the phase and the nature of risk involved in the phase of clinical trials that you are conducting will determine the nature of benefits you will promise. Okay? But again, when protocols are written and you have said that this ABC will be the benefit, we want to see that you will do everything by virtue of the resources and capacity you to deliver based on those promises. So clinical trials are expected to meet the priorities of research community. This is another critical point. And if I want to speak on this bullet alone, 30 minutes, 1 hour will not be enough. Because there's no way we can talk about this middle bullet without talking about what we call the principle of community engagement. The principle of community engagement says ab initio, even when you are putting your research ideas together, it should be such that those who are members of that research community, they are factored in the determination of the method and processes of your clinical trials. That's very, very important. That means the community where the study will be done, you need to factor their needs. They are the ones to tell you whether what you want to do or the approach you want to adopt is which is going to be what they feel as the most critical need for that hour, for that moment. So clinical trials must be scientifically sound and designed to yield meaningful outcomes. So in doing this, in ensuring beneficence, it means you need to factor in the role of gatekeepers, the role of community leaders, not those who are just claiming to be community representatives. You must assure that these are the genuine community representatives that people will hold credible when they claim that they want to represent the people. Next slide for time. I'm just rushing against time. Then we will have a fairly longer time to address some of these things. Or when we finish, when questions come. Oh, then above all, do no harm. But if somebody writes a protocol and says there's no harm in this study, that means that person didn't go to the school of ethics. That means that protocol should be returned to that person to go and learn more about what we call types of risks, levels of risks, and determinants of risk. In terms of talking about types of risks, we have physical risk, psychological risk, social risk, economic risk, legal risk, and there should be 5: physical, psychological, social, economic, and legal. Okay? But there's another risk that I'm beginning to see that, AI is generating, and which I agree, is talking about AI-related risks. Okay? And I'm being written in that area, and seriously, there are risks emanating from AI, even in clinical trials, even in academic activities, and even in therapeutic services. So, but our clinical trials should avoid unnecessary procedures and ensure safety protocols are in place. So, where you realize that your intervention or the procedure you want to carry out has a level of risk, the risk could be minimal risk, less than minimal risk, greater than minimal risk. And when we are talking about determinants of risk, then we are talking about probability and magnitude of risk. When we're talking about probability of risk, we are saying the likelihood as compared to the ratio or percentage strength, okay? And as found in extant literature and confirmed by experts in the field. But you cannot just document that. I also want to see that even if you have identified there are potential risks, I will ask you, what are the steps that you have put in to mitigate those risks? It's just like someone wanting to do a cesarean section, and you have the best surgeon in the house, but there's no midwife, there's no pediatrician. Even if the midwife or the pediatrician is there when you want to do your low segment cesarean section, you still also must ensure that you have a resuscitator. The resuscitator is the equipment with which we have a lot of components in there, things to suck them out, things to improve ventilation. That the baby is coming, from the CS, site. Less than 2-3 seconds, you're putting the baby on resuscitator, and you're ensuring that every bit about the life of the baby is assured. So also in clinical trials, if you're injecting a drug, make sure there are antidotes. That others have been injecting that drug and there have been no consequence doesn't mean that there can be no risk. I started from the domain of nursing, and I can remember in 1984, 1985, somebody was being given procaine penicillin right in my hospital. The needle had not been withdrawn from the gluteus maximum. The person reacted to procaine penicillin and collapsed and died. And the other experience that I knew was in those days of 84, 85 practice also, where I saw that someone was being injected with chloroquine phosphate for malaria. Okay, that was the drug of choice in those days. The needle was still in the buttock while this person reacted to chloroquine and also died. So that something looks subtle or simple doesn't mean that harms cannot follow your good intentions. Next slide for time. Next slide. So justice is talking about fairness in selection of participants. Okay? Justice is also saying you give to people based on their level of contribution. Okay? Don't discriminate in any form. But specifically when you have vulnerable population, you must bring in extra mechanism to ensure that they are not exploited. And that is why ethics committees need to be their advocates at that point in time, to ensure that they are not unduly exposed to risks and the benefits in studies are equitably distributed. Next slide. I'm trying to just round off now because of time. Okay, next slide, next slide, next slide, next slide. So what are the roles of the IRBs? That's IRBs in American context, but in Nigeria, Health Research Ethics Committee, HREX. They play a critical role in safeguarding the right, safety, and well-being of human participants. I may not say so much about this because a couple of those people who spoke before I came on mentioned some of these things, okay? And of course, because they are independent, they should also, with that level of independence, autonomously, without influence, review protocols, approve protocols, or monitor research proposals to ensure that those clinical trials comply with ethical standards and regulatory requirements. And what I mean by this, that means institutional, national, and international guidelines. Like institutional, for example, I am from the University of Ibadan, and I chair the Social Sciences and Humanities Ethics Committee for the entire university. Okay, so we have UI research ethics policy. So my review, the way by which my committee reviews protocols will also need to defer to that UI standard. That's institutional standard, okay? But apart from that, we also have national guidelines at the national level, and the national level 2007 National Code of Research Ethics, which is a subsegment of the National Health Act, that is the 2017 National Health Act, which was signed to law by President Goodluck Jonathan before he left office, becomes, even though it's not a full-fledged legal document, but because it's emanated from the National Health Act, which is a product of the federal legislature, it means the Nigerian code is supreme. It's different from the codes that I've seen in 17, 18 other countries that I've been to. And I do say this in any country I get to. What makes our code in Nigeria superior is that it's a derivative of a federal law. So even though we are talking within the domain of ethics, but our guideline is such that it can translate to what the court will, defer to in certain instances. So the function basically, as ethics committee members, is to assess risk-benefit ratio of proposed studies, to look at benefits and all those things that I said earlier. Let's move for time. Next slide, please. I'm just trying to run now. So ethics committee that tasked with protecting vulnerable populations such as children, also elderly, cognitively impaired individuals, scrutinizing protocols of fairness and additional safeguards, especially where vulnerable populations are involved. That protocol must pass through full ethics committee review. It cannot be expedited. It cannot be exempted. So ethics committees also ensure the scientific validity. And that's what I'm saying. A study that says, for example, that you want to test the efficacy of a drug, at the minimum, there's no way you will not need to have either a t-test, either an ANOVA, or a chi-square. A study that says it's determined, predictive validity of 2 IVs over a DV. There's no way you want to have the statistical analysis in such a study that will not have regression of various types. And of course, if you're now looking at possible factors that can impact between the independent variable and the dependent variable, then you are talking about moderators or mediators. And if you are talking about that, that means sequential equation modeling. And let me go into statistics. So next slide for time. Will have to be found in such a protocol. Next slide. I'm concluding now. So we need to have the authority to suspend. Somebody talked about this, so I will not reinvent the wheel. Next slide for time. But we need to hop our game when it comes to oversight of research activities at research sites. So in practice, principalism, when it's a principalism that talks about autonomy, non-maleficence, beneficence, justice, the guiding institutional review board, that is Ethics Research Ethics Committee, in evaluating trial proposals, ensuring ethical compliance before approval. And it also informs ongoing monitoring and post-trial responsibilities, such as providing continued care and access to successful treatments. And Research Ethics Committee needs to work with drug regulatory agencies. Like in Nigeria, the National Code of Research Ethics has subsections that defer to NAFDAC. That is about the safety and efficacy of clinical trials. But the NREC, of which I've been a member at the federal level for 11 years, will oversee activities related to the scientific strength and the ethical validity of studies. I may have to rest my case at this point. I'll be willing to have your questions. I'm done. So, thanks.
Thank you, Prof. Thank you so much. Please, can we appreciate Professor David once again? So we are going to have our last presentation now, and then we'll take the questions.
Okay.
So quickly, we also want to hear about regulation. So we have the NREC rep online. Mr. Do, can you hear us?
Thank you.
Okay, so please, you can go ahead and share your slides. We are sort of pressed for time, so basic guidelines, if you can help us in 15 minutes, please. Thank you.
Okay, can you see my, hello, good afternoon, everyone. Can you see my slide? Hello?
Yes.
Okay.
Yes, sir. We can see your slide.
Okay. So good afternoon, everyone. It's my privilege to also make this presentation, but I don't know why after the chair of the National Health Research the chair of the Gwagwalada Teaching Hospital, yeah, and my mentor and one of the pioneers of bioethics in Africa talked. I don't know what exactly you want me to talk about. After I'm sure Professor Adejima has already finished everything. So I don't know why you asked me to make this, a presentation. But anyway, since you say I should do, let me try to also make some presentation as you requested. The history of NREC. So as Professor Adejimo already said, Federal Ministry of Health. Anyway, before that, we know that research ethics was born as a result of scandal and tragedies that happened in the process of conducting research. And we are aware of the Nazi doctors' clinical trial during the Second World War. At the end, we have what is called Nuremberg Code. Also, in after the Tuskegee syphilis studies in Alabama County, also the U.S. have what is called the Belmont Report. Also in Nigeria, after the Trovan trial in Kano in 1996, we also have what is called the National Code of Health Research Ethics, which Professor Adejimo has talked about. So how did we arrive at this? So the Federal Minister of Health conducted the West African Bioethics Program, now Center for Bioethics and Research, to develop this broad-based document. And Professor Clement Adebamo and some West African faculty members in 2006 has drafted the national code, as Professor Adejimo mentioned. Also, this guideline presented to the Federal Ministry of Health and stakeholders across the country and later adopted by the Federal Ministry of Health. Sorry, next, okay. So as Professor Adejimo mentioned, the National Health Act provided to establish of the National Health Research Ethics Committee by the Honorable Minister, and the acts specify the composition, role, and responsibility, and NREC derive its power from this legislation. And if you look at Section 4, Part 4, Section 33 and 34 of the National Health Act, you can see this information. So, so what is NREC? So NREC is the ethics body which is responsible for provision of and ensuring adherence to the guidelines that govern ethical research practice in order to protect the human research participant in Nigeria. So the committee was an offshoot of the Dormant Ethics Committee, which had been in existence since the early 1980s. So what is the function of NREC according to the National Act 2014. So number one is to determine guidelines for the functioning of Health Research Ethics Committee. So that's why we have some guidelines. We have guideline of biobanking. We have guideline of involving research with people with dementia. We also have a guideline on animals on animal ethics. So these are some of the guidelines developed by NREC. So as one of these mandate, we also registered an audit ethics committee. We also set norms, NREC also set norms and standards for conducting research on human and animal, including norms and standards for conducting clinical trial. The committee also adjudicate in complaint about the function of ethics committee and hear any complaint by a researcher who believes that he has been discriminated against by an ethics committee. Also, the committee also referred to the relevant statutory health professional council matters involving the violation or potential violation of an ethical or professional rule by a researcher. The committee also institutes such a disciplinary action as may be prescribed against any person found to be in violation of any norms and standard or guideline set for the conduct of research under the National Code of Health Research Ethics. The committee also advised the Federal Ministry of Health and State Ministry on any research-related ethical issues. So these are the mandate of the National Health Research Ethics Committee as captured in the National Code of Health Research Ethics Committee, and also as spelled out in the what is called the National Health Act TV 14. So let's look at the highlight of this National Code of Health Research Ethics. So the National Code of Health Research Ethics empower NREC to register HREC, to also create a National Health Research, Ethics Committee database and categorize this ethics committee. If you can remember, the chair of the Gwagwalada Teaching Hospital mentioned that they are registered as category B. So NREC categorized this ethics committee. We have up to 5 categories of this ethics committee. We have what is called category A. So if an ethics committee is registered as category A, that ethics committee can review phase 1 clinical trial. The committee can review vaccine and genetic research. They can review all types of clinical trial from phase 1 to phase 4 clinical trial. But for now, it's only the National Health Research Ethics Committee that is operating as Category A Ethics Committee. Maybe in the future, we are going to have a regional ethics committee that can be registered as Category A Ethics Committee. But for now, it's only the National Health Research Ethics Committee is operating as Category A Ethics Committee. If an ethics committee is registered as Category B, that ethics committee can review phase 2 clinical trial. The committee can review vaccine and genetic research. The committee can review other types of researches. But if an ethics committee is registered as Category C Ethics Committee, the ethics committee can start reviewing phase 3 clinical trial. They cannot review vaccine and genetic research, but they can review like epidemiological research and other socio-behavioral researches. If an ethics committee is registered as Category D, the ethics committee can review phase 4 clinical trial. The ethics committee can review socio-behavioral research and other epidemiological research. If an ethics committee is registered as Category E, the ethics committee cannot review clinical trial at all. So how NREC categorize this ethics committee? NREC categorize this ethics committee based on the qualification, experience, and expertise of the members of the ethics committee in research, in science, and ethics, and also at the infrastructure available in the ethics in the institution and in the ethics committee. Also, the ethics committee, NREC look at the size. That's how many members the ethics committee has. Also, this is the basis NREC categorize this ethics committee. So also NREC also require all HREC members to complete NREC approved ethics training. So before we register an ethics committee, they must to train that ethics committee for at least 3 days on research ethics. And Professor Adejimo is one of our facilitators. Also, the Act clearly spelled the personnel and office requirement in establishing ethics committee, and the head of an institution should provide legal liability cover of the members of the ethics committee in case of litigation. So let the institution know that when a researcher, which is not common in our settings, they have to, when the researcher sue or a participant sue that ethics committee, it's the institution that should stand for that, ethics committee. And the ethics committee shall apply the initial registration, which is lasts for 2 years, and they have to apply for registration after that 2 years. So also the code highlighted about ethics mentioned that an ethics committee can charge fees. As you all know, NREC at the moment, we don't charge fees, but the code clearly stated that an ethics committee can charge fees. But when you go to other ethics committee, you have to pay for review. And also the code talks about the informed consent document, that it must be legible and not more than 8 pages. And most of the time, people are thinking that when we say 8 pages, they'll say it's too long. Some are saying that, but we say clinical trial, it should disclose adequate information. And if you can remember, Professor Adejimo mentioned about what are the key elements that you should mention in the informed consent. And you should make sure that the participant understands. So you have to disclose the basic information, but the code says it should not be more than 8 pages. But a protocol that is maybe 200 pages. We are talking about clinical trials. The protocol is 200 pages, and they say you have to disclose the basic information to research participants about that clinical trial. So I'm sure you know that the researcher will not find it easy to summarize the basic information about that in 8 pages. And this 8 pages is the maximum. Not that, even though the code did not talk about the minimum, but this 8 pages is what? Maximum pages. So I saw informed consent of 50 pages in Vanderbilt University. There was a time somebody submitted application to NREC. We saw 27 pages. I told him that, no, we can't accept this application because it's more than 8 pages. But what the code says that if you have additional information, you can put it on what is it called? Information sheet. So that's what the code says. Then ethics committee may offer consultation service to researchers, and NREC will exercise oversight function to ethics committee, and NREC and HREC have a disciplinary and compliance power on violation of the National Code of Health Research Ethics. So as an ethics committee, you can go, if you are monitoring what you approved and you discover that what you approved is not what the researcher is doing in the field, then you have the power to stop the study. You also have the power to suspend that study. Hello, can you hear me? Hello?
Yeah. We can hear you, sir.
We can hear you. Okay, all right. Okay. So, and you should know that an ethics committee should have, ethics committee membership must include a layman. Actually, what we are using now is community representative. But the WHO said that what we should put in our committee, we should stop using a layman. We should use we should stop using community representative. We should be using a layman, because you can get a community representative that is a professor of medicine. You can get a community representative that is a lawyer. So what they said is, what we should be using, we should be using a layman. So somebody that, if he doesn't understand informed consent, so that means there is possibility for a lot of people may not understand that. So that's why I put this community representative in bracket. So an ethics committee should have a lawyer. So in addition of possessing the professional competence necessary to review a specific research, an ethics committee should ascertain the requirement in terms of institutional regulation, in terms of also existing law in the country. So whenever feasible, you should involve somebody that is knowledgeable about that regulation. So whenever feasible, you should use a lawyer. An ethics committee should know that all the information that will come to the notice when servicing the ethics committee, or even after leaving the ethics committee, they must keep that confidential. So, and they should know that no member should participate in initial review or continual review when the member has a conflict of interest. And this conflict of interest is not necessarily essential, even for publication or even for recognition. So the researchers and also ethics committee members, they must to note this. And the code also includes clear process of full review, expedited exemption, and amendment of protocol. So as Professor Adejimo mentioned, an ethics committee, they look at the risks involved in the study. When the risk involved is high risk, and vulnerable population is high risk, that kind of study qualifies for full committee review and approval. And as we know, almost all the clinical trial, is a kind of study that involve what? High risk, because you are conducting research related to vaccine or new product or medicinal product. So if the risk involved is high risk, then the study qualifies for full committee review. And another one is expedited review. So when the risk is low risk, when the probability and magnitude of harm is not greater than that encounter in the daily life of all person or majority of the person you are going to involve in the research. So that kind of study for expedited review. So another condition is, even though the risk involved is low risk, but if vulnerable population are involved, like children, pregnant women, or homeless person, so that kind of study also qualified for full committee review. But if the risk involved is low risk or minimal risk, and there is no vulnerable population are involved, so you can consider the study for expedited review. So for instance, when you are what is it called, responding to a non-revealing or non-invasive survey questionnaire, at times that kind of study qualifies for expedited review. So another one is exemption. So exemption, if the risk involved is low risk and also no vulnerable population are involved, so it qualified for exemption. So what is the difference between exemption and expedited? So most of the time, when you are the studies that qualify for exemption, you see maybe there is no direct contact with the research participant, but the data, the research, you got the data from participant. So for instance, you are conducting research on existing data. You are conducting research on publicly available data, or you are conducting research on anonymized or de-identified data. So that kind of study qualifies for exemption approval. So for instance, when you are conducting research, just maybe to evaluate an institutional capacity to conduct research. So that kind of study most of the time qualifies for, what is called exemption. Okay. So they are saying that we have 5 minutes. Anyway, all right. So so these are the the composition of the members of the NREC. And so if you look at this picture, it's the first state of the ethics NREC members. In the middle, you can see is a Professor Eitai Lambo. Behind him is a Professor Clement Adebamo. So I'm very close to him. You can see is Professor Adejimo. So these are the first set of the NREC members, and some of them participate in the development of this National Code of Health Research Ethics Committee. So what is the role of regulatory agency? Is to review and approve application to conduct trial and to ensure the investigational medicinal product meet appropriate standard of quality, safety, and efficacy, and the trial participant insurance. So these are another issues we have. We recommend that before you conduct clinical trial or high-risk study, you need to get insurance for the research participants. But some of the insurance company we have in Nigeria, they will ask you, is it a life insurance? So these are some of the challenges. Some investigators, they will say, no, it's not life insurance. But I think it's a life insurance because you don't know what exactly is going to happen to the research participant. Maybe this research participant, because the high risk will be even death, or somebody will have a permanent deformities or disabilities. So the truth, as far as I'm concerned, is a life insurance. But some of the investigators, they will tell you it's not a life insurance. And also the regulators also conduct inspection of the research site to enforce compliance to regulatory requirement, and document facility record and any other resources related to the clinical trial. So more especially to monitor informed consent process, because if you look at some of the research that are being conducted in Nigeria, they don't get even, it's not informed consent they are getting. What they are also normally obtaining is just maybe they introduce themselves. The research participant will not even understand exactly that, look, he will not understand whether he's participating in research. At times they will be thinking that it's government that is providing something for them. No, let that research participant understand exactly that this is research to avoid any therapeutic misconception. So then what is the relationship between NAFDAC and NREC? So NAFDAC involved in trials involving drugs, device, and biologics, but you should know that NREC, any human research, NREC involved in it, or ethics committee involved in it. But most of the time, or most of the study, NREC, NAFDAC involved, they are either clinical trials or biological, study. And NREC also involved in all kind of, health research. So without the two approval, the study should not proceed. So the question is, who approval should be obtained first? Is it NREC or NAFDAC? Before NREC approved, before NAFDAC. But now we already have this agreement with NAFDAC. We encourage parallel submission because you can submit the application to NREC and also at the same time you submit the application to NAFDAC. And at times, if there is need, we conduct what is called joint review. So we, more especially for study, high-risk study that need expedited or fast-track approval. So NAFDAC and NREC need to, review that kind of study. So this principle of research ethics on Professor Adejimo already talked about this. So then reporting of adverse events. So occurrence of adverse event may reflect a change in the risk benefit of research. An ethics committee must therefore evaluate all adverse events. The objective is to ascertain whether the study should be continued, to ascertain whether the enrolled participants should be reconsented. And in multi-site study, according adverse event at any site should be recorded to all other ethics committee if it is a multi-site study. And in clinical trial, all adverse events must also be reported to drug regulatory agency. In Nigeria, we have NAFDAC. So the report should contain description of the adverse event in detail that allow an informed review of the occurrence, an explanation of why the event was unexpected, related to study and serious, and describe the changes to the research protocol, the consent form, and the need, if there is need to reconsent in order to minimize further risks of the event. And if no changes are proposed, then the rationale for continuing the study in the same way should be provided and update the risk analysis of the study. So I think all this Kudemo also talk about the community engagement. Actually, it's very important. All this already talked by Professor Adejimo. So community engagement is also very important, and it's one of the also ethical issues. Before you start your research, you need to go to the community and educate them about the study. And this will help you also in terms of retention of this research participant. Because when you help the research participant community make an informed decision, it will promote public trust and also retention of the research, participants. So community engagement is very important. And this community engagement, before you start your study, you're supposed to go to the environment. You're supposed to go to the community, and educate them. And if maybe you want to conduct research in your institution, after educating them, you can bring them to your institution and get their consent for them to participate in your research. So in conclusion, NREC is empowered by the National Health Act 2014 to regulate the ethical conduct of research in Nigeria in both private and public sector. So for new drugs and medical device, NAFDAC needs to give approval before clinical trial can be conducted. So thank you for listening.
Thank you very much, Mr. Ado Danladi. Please, let's appreciate him. I think with his presentation, he has answered some of the questions that people had raised regarding NAFDAC and NREC and the interface and what they felt was a bit of some overlap, but his presentation has addressed most of that. So we need to quickly go into a few questions. The time is spent, but we would take, I would suggest we take 2 questions in-house and then 2 questions online. I know there are a couple of questions online and some comments. We would address those ones because of time. Those we can't take now, we'll address them. And since participants are going to continue with the workshop, next week, Thursday, when we start the next session, we would upload those answers for those who had asked them to get to see what the responses were. So do we have questions in us? Okay, so please. We have 3. Okay, so we'll just take the 3 questions.
Thank you very much. I really want to appreciate Prof and the institution for organizing this workshop. And in doing that, I want to appeal. Having 100% is almost like a difficult thing to get. I understand that you said that you have to score 100% attendance to get certificates. I don't know if that is achievable, knowing that a lot of things can prevent you from missing a week, including network issues and so on and so forth. What I know that most people that organize this kind of thing, they will give you opportunity, maybe 48 hours or so, to go through the presentation and present your answers. So that qualifies you to having attended. Because if you are sick or network issue and you're not able to attend, that means you won't get certificate. So I think maybe we should review that that aspect. Then I wanted to ask you the issue about NAFDAC and HREC. First, I want to ask, as a clinician, can I initiate a clinical trial, or must it be a community contract organization who have to initiate that? If I feel that there's a drug I want to investigate on, can I initiate it? And going by that, what is the minimum budgetary requirement for one to go into a clinical trial? And there's not going to talk about NHREC, where you say if you are conducting research work in 2 or more than 3 states, you have to go national. But what information can you also obtain ethical approval from all the separate? Does it suffice to get approval from individual institutional ethical board than going for the national? So those are my questions. Thank you very much.
Okay, so thank you. We'll take the second one. Please, I hope the relevant people who are taking notes of the questions, Mr. Do, regarding the requirements for more than 3 sites across the nation, and then Dr. Beno Yakub for the one that has to do with the clinical trial being initiated.
Okay, thank you very much.
Yes.
My question goes to the NAFDAC Assistant Director. And I want to ask, I'm a little bit concerned about the proliferation of these ABA drugs that we see everywhere. You know, the Fafuron, so many T's that you see people displaying. Do they also pass through these clinical trials? And do you have, what are the regulatory guidelines given to all these people? Thank you.
Okay, can I? Okay, yeah. Thank you very much. I must say that I know that Professor Maxwell is going to bring the vote of thanks. So, but I really want to thank Dr. Beno and my chair of our institutional health research. They are doing a lot of work, on our behalf, and we appreciate them. So I want, now that Ado is online and Dr. Beno is online and the chair is online, there's a very important matter that we need to bring here, and that has to do with the CTA and the MTA. They've told us several times, in fact, the former chair of our NHREC said that this MTA is for your protection. But when we want to use it, we go back and forth with the sponsors. They don't want to use the templates that we give them. So we go back and forth, back and forth. And sometimes it drags for up to a year. You understand? Because you want to know that these samples that you're shipping, ideally, we shouldn't be taking any samples abroad. We should be doing, analyzing our samples here. But we don't have that capacity yet. So it's back and forth. What I'm asking, is there a way that our regulatory agencies can put their foot down, right, as to what we are allowed to do? Not just a suggestion, because the MTA from Tanzania is very rigorous, very explicit, very detailed, and you can't go around it. So that's one of the things I want us to kind of think. The other one is, everybody wants to get into clinical trial. So when you've gone back and forth with sponsors, and you finally agree to do a study, the way that what they consider fair market value in terms of what comes to sites, or the infrastructure that sites would have, or the kind of way that participants are reimbursed, even for transport and all of that, is quite different. Sometimes I think that Nigerians are the short end of the stick when you compare to other countries. And we feel that we are not being protected enough. You understand? Because it is the same study you're going to do. Someone in, in Europe, America, or even other African countries, they're having more support for their participants than we are having. You understand? So what the final one is, okay, I know that we get approval based on site readiness and facilities inspected, and sometimes they rent equipment and bring. At the end of the day, why do they have to take them away? Dr. Beno, we want you people to say something about this. If you bring a centrifuge, or you bring effusion pumps and you bring things, why do you want to take them away at the end of the day? I know it may be in some small print somewhere, but you don't have to take them away. You want to leave a site better than you met it. We want to be protected specifically in this way. I know we are signing all kinds of agreements, because sometimes if you delay, they will start the study and finish it even before you have arrived. Okay? So these are some of the issues. Thank you.
I think at this point we should get responses. So we'll start with Dr. Beno.
Thank you very much, Prof. and the rest of the people who asked questions. Again, let me start from question number one. You talk of maybe somebody wanted to initiate a study of his own. Absolutely, yes. You can do that. Again, we call it investigator-initiated studies. You can do that. You have the need to do it. But again, talking about the budget, I think you should know what you want. You do your sample size calculation, know your consumables, know the equipment you need to be able to actualize that study. I think the budget issue, it's yours. Right. So I hope I address the issue. Thank you, sir. Then coming to the issue of herbal drugs. Again, we all agree that it's a problem. You're talking of whether they go through clinical trials. We have one peculiar problem. You see, the federal government wanted to see how they can empower people, right? So encourage alternative medicine. In fact, globally, it's been encouraged. But the aspect of clinical trials, you agree with me that those who are the owners of those products cannot afford clinical trials as it is. That's the plain truth. But then what we do at the beginning, when they come, we ask them to prove basic human safety. Basic human safety. They do their safety analysis out there, and we confirm it with the animals to make sure that it cannot harm anybody. Then we've put a policy in place. If you register for 5 years, 2 years we give them, because we call it provisional registration, because they don't have clinical data to ascertain the safety or efficacy of whatever they are claiming. So if you look at the products that have gone through NAFDAC, they have a disclaimer that says these claims have not been evaluated by NAFDAC. And again, the product that has gone through NAFDAC will not tell you this heal this. Maybe for the management, but they don't claim healing, except those who have not passed through us. So we can do renewal for you twice. But we expect that at that moment you have started putting down some safety data, maybe collecting from the people that have used our products. Then we encourage them to come for clinical trials. Again, we've tried that a lot of times. Some of their worries is that people steal their intellectual property kind of, because if you give your formula, somebody who will do the research might. And of course, they have a reason to do that. If you remember one product they called Jobilin, right? They had that issue. I don't know whether they've even gone out of court now, because the person who founded that formulation, again, had no money, had no intellect to do that. So somebody came in to work with him. But eventually we started registering, then we saw a name change. And we began to ask why. So the man now came, and before he knew, the issue went to court. So those are the issues. They don't want to give out their formulation to the researchers because they are no researchers. So, but at a point, DG NAFDAC had to come in between and organize a meeting, a stakeholder meeting between people in the industry and those in the academia. Come together. Let's see what you can do. I'll start as a mediator. Bring in what you have. Let me give our guarantee that they will not steal your property. But I think we're still stuck in that situation as it is. But thank God we have the Natural Medicine Development Agency that is already ongoing, doing some, I think, discussion with some of these owners of the products and researchers. I think they are mediating between them now. Then I think we will be part of it and make sure that they do the right thing. Thank you. Then thirdly, the issue of MTA. I think I will allow Ado to talk about it because I believe they have a template for MTA. And as regulators, if that is our law, why should they refuse to obey our law? So I know other countries, regulators insist on MTA, but I think with us here, because we have MOU with NREC, we agree with what they are doing, asking for MTA, and they have a template, which to me must be adhered to. But Ado will answer that. Then, participant compensation, Prof. I think Ado too, this is an ethical thing. But again, for me, if you talk about giving our participants the same thing they give out there, again, you are trying to entice them or you are trying to induce them to, because if they are giving $100 in US, if you are giving $100 here, even me, I'll come to join the study. So you know that. So you gauge it based on the economy of where you're in the study. So if you're going to give $100 here, you're talking about 150 something thousand. Who among us will not want to go there? Oh, so that is it. So we try to avoid inducement and But then you should make sure you at least remunerate them as they come to give you information. Then we talk of site capacitation. Again, for me, Prof, I think you told me that time, honestly, if I am a researcher, for me to sign an agreement with you to do your study, don't even look at the money, but look at the capacity development, both human and site development. The sponsors are here. So if you are coming, some of the sponsors are here. If you are coming to conduct study at my site, and I don't have some of these equipments, and you decide to put them in, why should you take it back? That is part of your responsibility. I've seen sites that got cars, trucks, as we were doing the study, and after the study became theirs. It should have been decided. That is part of site development. So I don't see the moral justification for them coming to take it back. And for me, I don't think it's right. So I think Ado might be able to talk to the, okay.
Thank you for those lucid explanations. Of course, for retaining some of the items that have been used in the research, is tied to the capacity of the researchers in negotiation. It's not a must that you must participate in that research. Let me go straight to the point, the question whether if you have up to 3 centers, wouldn't you rather go to individual centers and get approval? No, that is not the rule. Each of the centers you'll be going to, of course, requires to give you, to domesticate the approval you have brought from the NREC. After all, NREC does not charge you any fee. If you are sure of what you are submitting, advance it to them, they'll look at it only and then gives you the approval. So when you are applying to each of these 3 stations or more, you just give a copy of that into it. They knew that you have fulfilled the righteousness, you have gotten the approval from the higher authorities, and then we'll domesticate it and see whether it's suit our condition or otherwise. Getting the NREC approval does not necessarily mean we must give approval to you. If we look at the content of your submission and the nature of our environment, we need to domesticate it appropriately. Thank you so much.
Thank you. Thank you for the responses. Mr. Do, can you help us with the issue of the MTA? I know, let me just give a bit of background. I know that there was something posted on the platform about genetic material. That's still related to the MTA. Sometimes, as part of the studies, they want you to sign off on that. They can use maybe the dry blood spots, genetic material, and all that for future studies. I know we've had like 2 studies where the PI said, no, I'm not consenting to that. So please, can you respond to the issue of MTAs and then the fact that they want us to modify the template that NREC has in some situations? Please unmute yourself and okay. Hello, Mr. Do, if you can hear me, we can't hear you. Please unmute yourself.
Yes, I can
Okay.
I can hear you. So
Okay, we
thank
can
you
hear
very
you
much.
now.
But before I say anything, please can you unmute Professor Adejimo? So can you also unmute Professor Adejimo, please?
I think that's been done. Yeah.
Okay. So Professor Adejimo. Hello, sir. Okay.
Hello, how are you?
Okay, I'm fine, sir. So
Yeah.
I said maybe if you can respond to some of the questions related to material transfer agreement and also the multi-site approval. I hope you had the question, sir.
Well, straight to the point, when it comes to MTH, it's a global standard. I overheard the one of the commentaries saying that in Tanzania, it is enforceable and you can't go around it, if I can quote the person rightly. But I need to say, whether in Ghana, whether in South Africa, whether in New York, MTA standards remain MTA standards. They are to ensure that items that are obtained from clinical trial sites or experiments that are to be shared with other centers or other locations, other countries, that there is let me just continue, that there is the need, but I hope I'm audible anyway. I hope somebody can hear me.
Yes, sir. We can hear you,
Okay,
sir.
good. So it's to ensure, it's part of the procedures to ensure that people are not unduly exploited based on their location, based on their vulnerability, based on other standards. Okay? And so MTAs is to say, the samples that you are taking, this is how much of it you can use, and this is the use you can put to it. And if we don't do that, we will run into what we call neo-colonization, whereby people will still come in this century to still exploit us as we are citizens. You cannot just obtain samples from one part of Canada, shipping it to the US, and you think Americans will use it anyhow without Canadians raising a red flag. And what is good for the geese is good for the gander. That's what we'll talk about, the difference between equity and equity and equality. They sound similar, but they are not similar. They are not perfectly similar. So what we are saying is, to maintain justice, we need to respect the values, the standards, and in this instance, the products, the samples that have been obtained from one part of the world, and to limit the extent to which that sample can be put to other uses, apart from the use that are in the development of the protocol. How much of it you can use, how you can use, all those things must be decided at the point at which studies are being put together, especially when it comes to multi-site clinical trials. So even though the host nation most times will be the one where the grant is coming from, but that does not give them that power to coerce, to colonize, other nations or other centers where similar studies are being replicated or conducted. And there are very many examples, if you remember also. I guess the issue of the HIV clinical trial drugs in those days also come to mind, whereby they will come here, they'll take samples, and they put samples to uses beyond what they claimed they wanted to use those samples for. And of course, that also leads us to the question of 80-20 divide. The products are accessible to them, but they leave only 20% of the accessibility and affordability to us. The 80% of products that they extracted from us now, of better accessibility and better affordability to countries where these specimens are shipped to. One of the presenters also said that if we talk about $100 in terms of inducement and compensation, where that he will participate, that's also understandable. If you allow people to have unfettered access and utility to samples that they are taking from elsewhere, that would be unethical. And we are saying in our time, no. I want us to read the publication of people like Solly Benata. Solly Benata is a South African Canadian. He's written extensively on the African perspective of, sharing of research gains. Okay, so you can download the papers written by Solly Benata, especially papers written around 2000, 2006. He's written extensively on some of these issues. But my own take-up point is, my take-home point is, whatever is good for the geese is good for the gander, and people cannot, in one way or the other, download. Do I say overburden or extract beyond what is necessary of what their normal call should be when it comes to exchange of items at research sites? So that's my position. There's another issue that you raised now, which I cannot remember. So in terms of material transfer agreement, it must be legally documented. And this is why you need lawyers when you are developing such documents. Don't just go into it saying you are a clinician. You need lawyers that are well wired when it comes to international laws and international exchanges. So again, please go and look at CIOMS, Council of International Organizations of Medical Sciences. It also specifies what the standard should be when it comes to material transfer agreement, especially the 2016 version of SIOM's guideline. It specifically mentioned that the use to which it was agreed was, is the use to which the host nation that is taking samples from other countries should put it to use. They shouldn't go beyond what they claim they want to use that sample for. If you have any other excuse, there must be a fresh consenting process. And the need for fresh consenting process was why Tindana, Paulina Tindana and his group, and our group, is a Ghanaian, Paulina Tindana from Ghana, DeVries. These are the two people who came together to write a paper. And what did they write on? They talked about informed consent in multi-center studies. So they also talked about the issue of sharing of genomic samples. When genomic tissues and samples are taken from Africa to other parts of the world, how much of it can you use? Should be rested on the agreement, the consent that the community from where that sample was taken said you can put it to use. And it's the country where that sample was taken that has the right to, through the regulatory agencies, in this case, NREC, to put their feet down, to develop the guideline, and to enforce, the utility of the sample at the sites where they will be shipped to. That's my position. Thank you. So NREC need to do a little more in terms of enforcing and putting their feet down. If there's the need for us to modify our document for, MTA, we may need to do that. I know also there are countries with better developed MTA documents. Thank you. Over.
All right. Thank you very much, sir. Sorry, before you talk, let me add something to please. Can you hear me?
Okay.
Okay, thank you, sir.
Go ahead, please.
All right, sir. So concerning this material transfer agreement, actually, as Professor Nadu mentioned, we have a template, and as also Professor Amos, we have a template, and you know that before you ship out any biological sample, NREC has to endorse that. We have a template. The provider scientist has to sign that document. Yes. The head of the institution has to sign it. The recipient scientist has to sign it. Also, the recipient organization. And the template is detailed. You tell us how are you going to ship out the sample.
Yes.
How are you going to discard? It's detailed.
Yes.
But any person that doesn't want to, use that template, we cannot endorse it. And you should also explain to that person that there is no need, must we conduct a research? So without that, we are. But the issue is this: there was a time they shipped out biological sample from Irwa.
Let me even tell you, in University of Ibadan, we also have a very robust technique of enforcing MTA standards for samples that have been taken within the institution, University of Ibadan. We also have a guideline for that in UI.
Okay. Thank you, sir.
But we defer to the national body. That's what we put in UI guideline. That apart from US standard, we should still enforce the national standards.
Okay, thank you, sir. But the issue is this, sir. At times, when they ship out the biological sample, you can even discuss with your sponsor that he wants to go and monitor what they are doing with the sample abroad. Because recently, Irwa Specialist Teaching Hospital, we have even developed a checklist on what are we going to monitor. So we went to Hamburg twice to see the sample on Lassa, fever research. The sample they shipped out to the, Germany. We went there and tracked those, sample. So if also the Wagwalada Teaching Hospital, you can discuss with your sponsors through the ethics committee in your institution, and you inform NREC. We have to go and see what they are doing with the sample. We went to Hamburg, but another issue is when you get there, they will tell you this is a biosafety lab level 4, you cannot enter. All the sample that you shipped out, they will just show you on paper and on the, on the laptop or on the computer. So, but at least there is this, we went there, we developed a checklist. And in addition to that, now there's what you call DTA. If you are going to ship out data, you must also submit that to NREC for endorsement, data sharing agreement. Because the way you are taking sample, or data, things are changing. But what I was also want to add to, you should know that apart from human sample, that is what we call Nagoya Protocol. Nagoya Protocol, if you want to shift out maybe animal sample, or that are not human, you have to have that Nagoya Protocol, and you have to contact Ministry. They have that Nagoya Protocol. It's also similar with the material transfer agreement. So concerning the multi-site study, as the Professor Issa, the chair of the Gwagwalada Teaching Hospital Ethics Committee mentioned, yes, multi-site, even if it's one clinical trial, it's only NRED that will approve that. But if it is a multi-site in 3 sites, let's say in 3 different states, in different geopolitical zones, let's say Abuja, Ibadan, and Kano. So these are 3 sites in 3 different geopolitical zones. You can come to NREC, but it's not compulsory. But you can come to NREC, we can give you approval. If you want to go to the site, specific site, and get it, fine. But it's not compulsory. But Gwagwalada Teaching Hospital cannot give approval and go and monitor what is going to be conducted in Ibadan. But NREC has the mandate to monitor all the research. So we are even helping the researchers. It's not that we are trying to make it difficult for them. That's concerning the multi-site study. Then the other one, this collaboration research, we know that one of the collaboration is ethical for you to build the capacity to even provide infrastructure to the institution. I don't see any reason if maybe you provide a machine, you provide something at the end, you go and take it. I think how ethical that is. You build the capacity. In fact, that's why at times we don't want them to even ship out the biological sample. Let them bring the machine, train our people here so that we'll be using that. But I don't know how ethical that is. All right. And for issue of the undue inducement, as Prof also mentioned. So there is undue inducement, there is also inducement. So inducement is ethical, but I think undue inducement is not ethical. You can incentivize the research participants. You can encourage them to participate in your research, but you should not give them what is called undue inducement. So thank you, sir. Thank you for listening. Bye.
Thank you very much for the explanations. Professor Nodo has something to say.
So I know that the leaders of thought on this program, particularly Professor Dejumo, the NHREC people. So talking about inducement and what has happened in the recent past in terms of being able to look after subjects or participants in human research within the country and in Africa. You know that there has been some withdrawals of some, drugs, right? Because people died from malaria, right? In, just normal malaria that people have. And that actually affected that product. So what are we saying? Is our participants from this part of the country need a little bit more support in order to come to the hospital, in order to eat well, in order to participate in clinical trial, in order even to access care, you understand, and routine drugs. They need more help than others from high-income countries. We have to be adaptable. We have to provide for these people. Otherwise, if you say, okay, it's the same thing as everybody, you can get results from that system that will mess up the entire clinical trial. So please, let's be careful about our definition of inducement. We have to provide a kind of level playing ground to allow participants from here to participate in a clinical trial. It can't be the same thing. You understand? You cannot say it's an inducement when the person is not even feeding well. The person cannot eat well, and you want them to go and come live their, we just have to redefine what we mean by inducement, with the kind of economic conditions that people are faced with. How do they get the standard of care? If when standard of care is being provided, insurance is being provided, all the tests are being provided outside for other people, and these people are the ones paying out of pocket, and you want to redefine, we have to look at it realistically. Honestly, Professor Adejumo, we have done our national code. That's many years ago. Let us revisit some of those things so that we give our participants maximum opportunity to partake in studies. Thank you very much.
I think we should let's appreciate Professor Nodo for that, because if we were to start sharing experiences from what we've seen since we started some of our trials here, this is a very important topic. For instance, we're in a hospital that, if you want to qualify it, you won't really say it's in a cosmopolitan place. It's far away from town. You won't compare it with maybe some patient who is maybe in Ibadan, close to UCH. We've had instances where it was raining. We had participants who were going to max out on their window in terms of visit and needed to show up. And the person is coming from somewhere like Suleja or Karu. You say, no, please, can you take a boat? Because you need to be here. And you're acting within very short time periods. For instance, those samples must get to the airport before a certain time, before like 1 p.m. Otherwise, customs won't clear it for it to be shipped abroad that day. And you say, take a boat, come wherever you are. And the person comes to and fro boats. Whatever stipend you're giving, $30 is gone. At the end of the day, with all the stress.
Hello, let me just come in there. Let me come in there.
Yeah.
Hello.
Yes,
it's unfortunate this meeting may not afford us the opportunity to actually dissect the nitty-gritty of some of these issues that we are raising. But what you are saying is within the ambit of what we call reimbursement, compensation, and inducement. What you are speaking on specifically addresses what we call reimbursement. The literature is very replete with the need for us to be mindful of circumstances and situations such as the one you have proffered here. And in such a situation, we're being encouraged that the concept to adopt or to deploy is what we call reimbursement. So even though we have said everybody should come from Gwagwalada to National Hospital at the cost of 5,000 to and fro, but because it's raining, may not need to wait for a taxi and all of that, is now spending about 7,000 or 10,000 naira. You still encourage that person to come, but the extra amount of 5,000 on top of the 5,000 you are giving to every other person is what we call reimbursement. Ethics recognizes that globally. Thank you.
Okay, so thank you very much, Prof. I guess this is one discussion that will continue. We have a place in the course, in this 12-week course, that is talking about budgeting, patient reimbursement. So we'll continue this discussion. And then the good thing is that a number of sites on SparkNet are also involved in clinical trials, and we want to share experiences, practical experiences of what's been happening. So, and we can, at the end, be able to maybe reach out to NREG to look at some of the guidelines that they already have ongoing. Okay, so thank you very much. I want to appreciate everyone. I know there are questions, about 3 questions online. Please, I want to appeal. I think about 1 or 2 of them have already been addressed. Please, we would answer those questions, and you will get to see the responses. The time is fast spent. I want to call Mr. Okay, before I call on Mr. Tayo to give the vote of thanks, Professor Nodu will come and give us the summary notes, especially with regards to the incoming weeks and other things, and then Mr. Tayo will give us the vote of thanks. Please, we want to appreciate everyone. Professor Dejumo, we really appreciate you. Mr. Danladi, we appreciate you, and every other person that's been part and parcel of this. Thank you so much.
Okay. Thank you.
Yeah,
Bye-bye.
Thank you very much. I can see that the numbers online have kind of remained about the same, through the day. And that just shows that this is an area of felt need. So I'm sure that Mr. Tayo will come and give the vote of thanks, but just to kind of tell us, we've had a very tantalizing day, morning and afternoon of just talking about the introduction to clinical trials and the phases of it, the history and evolution of clinical trials, the stakeholders, and then we had a press briefing, and then we went on to talk about the ethics and the human subject protection and having our regulatory agencies talk to us. This has really been amazing. And I'm sure that next time we are doing the GCP, we'll have echoes of today's discussion, with us. So for the next 11 weeks, we are going to be considering different aspects. We want this course to be practical, and we've chosen faculty who are actually performing clinical trials so that we're able to share our experiences and be able to actually do better than what we are doing. I met someone from Egypt some time ago, and she said, you need to be firm when you're discussing with sponsors if in terms of budgeting. And honestly, this is a case that we will want to dwell in detail when we come to budgeting, because sometimes people are not having the best that there can be in that negotiation process. So we have week 3 will be clinical trial documentation. And this is actually very, very important that we learn. In each of the partner sites, we have research assistants and we have data clerks. We need to be taught how to document. Once you start participating in clinical trial, there's a level of documentation that is required even for your routine work. And we are going to learn, how to do that. And then, of course, the managing the document, or the different software regulatory, clinical trial regulatory software that we are, conversant with, and we use, in the course of the study. Then we are also going to consider challenges of managing multi-site studies and the guiding principles of ethics and clinical trial preparations, the protocol, the importance of protocol, what not to do when you have a protocol. You don't, you're having a study, and then at the end of 1 year, you decide to do something differently. You're not allowed to do that. Protocol deviations are a big red flag. And so we are going to learn how to really prepare ourselves. And then through week 8, we have actually implementing. We are going to talk about site selection, feasibility, how to prepare yourself, how to qualify, reporting standards, all of that. You can see on page 3, so that I don't go on and on. We have the entire course, we have, the course outline written on page 3. So you can follow through. But the important thing is that it's 100% attendance. 100% attendance for you to get the certificate. I think it was a week ago, I kept getting email notifications that I was supposed to go and do some training in a trial that we had already completed it. And I was wondering, why are they disturbing me? They kept saying, go and do this training, go and do this training. So I went in and I did the training, but I enjoyed every aspect. There were things, it's GCP. Every 2 years you're doing GCP. This was another GCP by this sponsor, but I enjoyed doing that. I failed one question, and they say you failed, so retake the test. So I retook it, and of course, I passed. So that was 100%. So every aspect of what we are talking about is important. And before you get this certificate, there's a pre-test and a post-test. But even if you pass the test and you did not attend all of the sessions, you will not get the certificate. Just so that we are on the same page. Why is it important? Because clinical trial, there are ethical, there are regulatory and legal implications of giving IPs to people and monitoring them. So we must be aware, and we must hold ourselves to a high account of a high level of responsibility before we go into these processes, or even as we continue. So it's 100% mandatory as before you get the certificate. Thank you very much.
Yeah, good evening, all. I think it's getting to evening now. And it's already evening, right? You know, we plan to stop at 3:15, so, but the energy and the engagement has brought us up to this time, 4 p.m. We want to thank you all very much for your attendance, those online and those on site. On behalf of SESTA and the University of Abuja, we thank every one of you. Thank you for those who have traveled in to be at this meeting today. Let me thank our regulators very much. NAFDAC, Dr. Beno, HREC, Professor Issa, NREC, Mr. Ado Danladi. Thank you for what you have shared with us. The faculty, you know, what do they call the generation after Gen Z? Is it Alpha, right? Okay, there's something they say those who know, know. Thank you for the faculty. There are people who know. And they've shared with us. Again, Professor Nodu, Dr. Kwaku, thank you, traveled in from outside the country. Yes, so Dr. Kwaku, Dr. Frida, joined in from Tanzania, Kenya, okay. Thank you. Of course, again, Dr. Beno Yakubu from NAFDAC, Professor A.Y. Issa, UATH HREC, Professor Emeritus, or is it Emeritus, Professor Ifeoma Okoye, thank you very much. Set the scene in the morning. Professor Adebayo Adejumo, you woke a lot of us up when you are speaking this afternoon. Thank you, sir. And for everyone here, we thank you for all of your contributions. And please remember that this is just weeks 1 and 2 of a 12-week course. You have to be part of it all through the other 12 weeks. It's on Thursdays, and it's at 7:30 p.m., 1.5 hours. There is time for each of us to be able to get back home, hopefully, and then connect virtually. Thank you, sir. Have I missed anybody? Sorry? Oh, okay. Yes, he said I missed myself, so I'm thanking myself. Thank you, sir. There's lunch. It's at the back there. So we would like the sponsors. Oh, I forgot the sponsors. I knew I was missing somebody. How can I do that? Yes, and thank you very much to our sponsors, IQVIA. We appreciate this opportunity to partner with you. And also to learn a little bit more about at least your space and what you do. I particularly took a lot out of the stakeholders that Dr. Frida shared with us. So thank you for that. I hope after these 12 weeks, like Prof said, there will be further opportunities to really engage. Of course, BVGH, who also are part of those supporting us in this program. Thank you very much. Now, if I'd left out the sponsors, it's very likely that, let me not share that. But thank you, we appreciate you. Please, lunch is served at the back, and we'd like our sponsors and faculty to please go first. And enjoy the rest of the evening, and see you Thursday next week. That will be 25th of September at 7:30 p.m. West African time. Wherever else you may be, God bless you.